Hello. Hello, check. Hello.
सुप्रभात सभी को। जैसा कि कल हमारे यहाँ 2 दिवसीय सैटेलाइट कॉन्फ्रेंस की शुरुआत हुई और आज उसका सेकेंड डे है। कल के जो शेष बचे हुए सत्र हैं उनका प्रेजेंटेशन और हमारे बीच उपस्थित विभिन्न राज्यों से आए विभिन्न विधि एक्सपर्ट्स अपनी विचार यहाँ पर रखेंगे। तो सत्र की शुरुआत करती हूँ। चूंकि हमारे माननीय पुलिस महानिदेशक और साथ ही हमारे अपर पुलिस महानिदेशक भी आज इस सत्र में उपस्थित हैं, तो मैं पहले उनका स्वागत करती हूं। अब आज की शुरुआत करने से पहले कल के पूरे दिन के कार्यक्रम की मैं एक रूपरेखा आप लोगों के सामने उपस्थित करती हूं। कल इस सत्र का विधिवत आरंभ हुआ। इस कॉन्फ्रेंस का हमारे प्रभारी निदेशक के द्वारा ये बताया गया कि बिहार में पहली बार 1983 में फॉरेंसिक साइंस की कॉन्फ्रेंस प्रथम बार आयोजित की गई थी। इसके बाद अब यहाँ पर कॉन्फ्रेंस का हमने शुरुआत किया है। इसके साथ ही हमारे माननीय पुलिस महानिदेशक के द्वारा बताया गया कि विधि विज्ञान प्रयोगशाला की बिहार में शुरुआत से लेकर अब तक की जो उपलब्धियां रही हैं उनके विषय में अवगत कराया गया और भविष्य में अनुसंधान के क्रम में विधि विज्ञान की महती आवश्यकता पर प्रकाश डाला गया। साथ ही यह भी बताया गया कि नए कानूनों के लागू होने के उपरांत पुलिस अनुसंधान में कांडों के उद्भेदन हेतु फोरेंसिक विशेषज्ञों की सहायता लेने में बिहार राज्य भारत में पहला राज्य है जिसमें कि लगभग अब तक 18,000 कांडों के उद्भेदन में विधि विज्ञान की एक्सपर्ट्स की सहायता ली गई है। और इसके साथ ही हमारे अन्य चीफ गेस्ट डीएफएसएस चीफ श्री एसके जैन के द्वारा पूरे भारतवर्ष में विभिन्न राज्यों के और विभिन्न केंद्रीय विधि विज्ञान प्रयोगशालाओं में होने वाली अनगिनत महत्वपूर्ण उपलब्धियों की जानकारी दी गई। आने वाली समस्याओं के बारे में अवगत कराया गया। साथ ही उनके समाधान के विषय पे चर्चा की गई और इसके उपरांत विभिन्न राज्यों से आए हुए विभिन्न विधि विशेषज्ञों के द्वारा उनके किए गए शोध कार्यों पर प्रेजेंटेशन दिया गया, जिसको कि हमारे कई माननीय अतिथिगणों ने मंच में संचालित करवाया।
सच कहूं तो कल के हमारे विद्वान जनों के वक्तव्यों से एक ऐसा सार प्राप्त हुआ कि एज ए फॉरेंसिक एक्सपर्ट हमारी ड्यूटी बहुत ही बढ़ गई है। पहले ऐसा कहा जाता था कि 100 दोषी छूट जाए मगर किसी निर्दोष को सजा नहीं मिलनी चाहिए, लेकिन अब मैं कहूं कि अब यह परिमार्जित होकर इस प्रकार हो गया कि कोई दोषी छूटे भी नहीं और निर्दोष को सजा भी न मिले तो कोई अतिशयोक्ति नहीं होगी। इस प्रकार एक फॉरेंसिक एक्सपर्ट की कानूनी रूप से नैतिक जिम्मेदारी बढ़ गई है, जिसके लिए हमें समयानुकूल पॉलिसी बनाने की आवश्यकता है। इन्हीं सब पहलुओं को अपने में समेटते हुए इस सत्र का थीम है लीगल, इथिकल एंड पॉलिसी एस्पेक्ट्स, जिसके प्रारंभ के लिए मैं डॉक्टर ए.के. शर्मा सर, डायरेक्टर सीएफएसएल कामरूप को सादर मंच पर आमंत्रित करता हूं। सर
डॉक्टर ए.के. शर्मा के बारे में अगर मैं एक संक्षिप्त परिचय दूं तो डॉक्टर ए.के. शर्मा ने अपना पीएचडी पंजाब यूनिवर्सिटी से कंप्लीट किया और इन्हें विभिन्न प्रकार की छात्रवृत्तियां भी मिली। सीएफएसएल कोलकाता में इन्होंने वरीय वैज्ञानिक अधिकारी के रूप में 1994 में बायोलॉजी डिवीजन में अपना योगदान दिया। उसके बाद इन्होंने कोलकाता सीएफएसएल में 1997 में डीएनए यूनिट की स्थापना में अपनी सक्रिय भूमिका निभाई। इनके द्वारा नेपाल और भूटान सहित महत्वपूर्ण डीएनए मामलों में जांच की गई और रिपोर्ट प्रस्तुत की गई। इसके बाद इनके द्वारा सीएफएसएल गुवाहाटी का विस्तार किया गया और इन्होंने एक समय में सीएफएसएल कोलकाता और सीएफएसएल गुवाहाटी का निदेशक के पद का दोहरा प्रभार संभाला। सीएफएसएल गुवाहाटी की इंफ्रास्ट्रक्चर में इनका काफी योगदान रहा है। इन्होंने डिजिटल फॉरेंसिक्स, डीएनए, नारकोटिक्स, विस्फोटक, रसायन विज्ञान और मनोविज्ञान सहित विशेष विभागों की स्थापना में अपना नेतृत्व दिया। इसके साथ ही इनके कई सारे अनुसंधान एवं प्रकाशन हैं, जिसमें कि इनका रिसर्च थीसिस पर इनका ऑब्जर्वेशन किया गया है। साथ ही साथ कई राष्ट्रीय और अंतर्राष्ट्रीय स्तर की समीक्षित पत्रिकाओं में इनका शोध प्रकाशित हुआ है। समय समय पर इन्होंने विभिन्न वैज्ञानिक पत्रिकाओं के लिए तदर्थ समीक्षक के रूप में कार्य किया है। विभिन्न न्यायालयिक अकादमीज में, पुलिस अकादमी में, चिकित्सक संस्थानों में और विश्वविद्यालयों में इनको व्याख्यान देने के लिए आमंत्रण किया जाता है। अब मैं श्री मतलूब रजा खान सहायक निदेशक को हमारे गेस्ट का वेलकम करने के लिए मंच पर आमंत्रित करती हूं कि वो आए और उन्हें पौधा देकर सम्मानित करें। श्री मतलूब रजा खान। अब मैं श्री हिमजय कुमार, डायरेक्टर इंचार्ज एफएसएल पटना को मंच पर आमंत्रित करती हूं एज अ गेस्ट स्पीकर फॉर सेशन 1। श्री हिमजय कुमार का स्वागत करने के लिए मैं मोहम्मद मुश्ताक अहमद, सहायक निदेशक बैलिस्टिक को आमंत्रित करती हूं कि वो मंच पर आएं और श्री हिमजय कुमार का पौधा देकर स्वागत करें। Now I would like to Mr. Ruslan, our Russian delegate, please come on stage for a guest as a guest speaker. Now I request to Mr. Deepak Kumar, Assistant Director, please come to the stage and welcome Mr. Ruslan by giving him a plant pot. सत्र की शुरुआत करने से पहले मैं श्री डॉक्टर ए.के. शर्मा सर से अनुरोध करती हूं कि वो मंच पर आएं और इस कॉन्फ्रेंस के बारे में और अन्य बातों पर अपने विचार रखें। सर।
Respected DDGP, dignitaries on the dais and dignitaries and ladies and gentlemen, I have joined today, but I have seen this conference proceedings briefly, even yesterday also, right, on the mobile and the online also, right. So there were some very good presentations also, right, and I would like mainly especially mention the presentation by Dr. Hirak Ranjan Das. That was perhaps the best presentation we ever had, right? So we have a lot of things to learn, and in fact biologists or the DNA scientists should have a forum. Perhaps this is the best beginning and we should continue it annually, right? I'm grateful for the organizers for calling us, all of us, to have this interaction and the discussion, right? I hope it will be very fruitful and will carry the message to our departments or the organization. Thank you very much.
थैंक यू सो मच सर। अब मैं सत्र को आगे बढ़ाती हूं और जैसा कि आपको आज के सत्र की थीम के बारे में बताया गया है, तो अब मैं श्री हिमजय कुमार डायरेक्टर इंचार्ज से रिक्वेस्ट करती हूं कि वो आए और उनके विषय पर प्रकाश दें, जैसे कि हम सब अभिभूत हो सकें। श्री हिमजय कुमार।
Respected chairperson on the dais and the guest speaker, and respected DGP sir, ADG sir, Dr. S.K. Jain sir, Dr. Sharma sir, Dr. Mohapatra sir, Devendra ji, my colleagues, and other dignitaries from CFSLs, FSLs, scientists and students. Today I got the opportunity to deliberate on uniform report writing, recent advancements and future aspects. This topic was regularly been given to me by Mohapatra sir. Always he was focusing that please you carry on this. And sir, I would like to mention that earlier also when I was in the committee sir, made by you sir, Jain sir, this topic was only given to me. I had worked on it sir, and it is very great opportunity for me to deliberate here on this. Sir, before starting, we all DNA experts who are working in the laboratories, we faces many challenges in court. Basically, whatever examination we carry out and the challenges we face in the laboratory, that can be sorted out by our end, by technicalities, involvement of technicalities, removing hurdles. Really, it was very great presentation, sir. Sharma sir has already mentioned. Dr. Hirak Ranjan Das sir, he really presented very well, and he actually given us a way that we must think on it. Rather of just applying different technologies and we are slave of that, being a scientist, it is our bound duty that we must come over it. We must find some way. Although today government is spending so much on us, giving us sophisticated instruments, we must amalgamate. I also thank sir, Dr. Switi Sharma has also shown sir yesterday that her work on ATR, FTIR for differentiating the PM blood and the peripheral blood as well as the menstrual blood. And for that advancements also, I have given suggestion to her that you please also go for histological examination to validate your that particular thing, so that you will get a controlled method, and then we can devise that method and we can get that method in our laboratories. So sir, actually I have mentioned that why uniform report writing is required. Sir, I have a judgment, sir, of the Honorable Madhya Pradesh High Court, Honorable Jabalpur High Court. It is related to DNA examination only. Recently, sir, Dr. S.K. Jain sir had shared me the video also of the courtroom, and I went through, sir, that. Why? Because it clearly gives us insights that why we need uniform report writing. It was not the fault of the expert, but rather the things were misinterpreted by reading the report. So if the report, uniform report should be applied to everyone, and we can have a similarity, and we must have a SOP that what we should add in the report, what we should not write in the report, how should we present our report before the court of law. Because sir, many times I also have seen that despite of working and giving proper report, the things changes. Why? Because experts could not present themselves properly in the court. They fails in explaining the report before the court of law. Although content is there in the report, but they fails in presenting that. So if uniform report writing will be there, all experts will feel easy and comfortable in the court of law that how to interpret the things, and the court will also not face challenges. Sir, in this particular case, what happened that we can read, sir, that the court has written, sir, we are not taking a final call today, awaiting reports from the Forensic Science Laboratory Hyderabad and Forensic Science University Gujarat. But what we had noted and what has been submitted before us is material. In the present case, the victim is one, we will call her, sir, I have hidden the name, we'll call her X, and there are two accused persons who will be termed as A and B respectively. Exhibit P34 is the report from the State FSL Bhopal DNA fingerprinting unit on dated 25/11/21, which is reproduced as such for the proper appreciation of age and every word of the report, which will be material while we proceed with the discussion and our findings. Similarly, the exhibit 38 is the report dated this co-appellant. Both the reports have been generated by scientific officer and assistant chemical examiner, regional FSL Bhopal. Report on page 1 of the report, consisting of 4 pages, deals with the sample numbers assigned to, although we mentioned the exhibit details on the first page, obviously, sir. Also consists of a cover page signed by Joint Director as it was dispatched. Now on page 2, what it is mentioned that, sir, Y chromosome STR DNA profile results have been mentioned, and it is mentioned that uninterpretable low Y chromosome STR DNA profile was obtained from the underwear, vaginal slide, and pubic hair of the victim. Whereas alleles obtained from the blood sample of accused in the first column, genetic markers have been given. In this report on internal page 2, it is clearly mentioned that since uninterpretable low Y chromosome STR DNA profile was obtained from the vaginal slide, underwear, and pubic hair of the victim, therefore it cannot be matched with the profile obtained from the blood sample of accused. Now, the result of autosomal STR DNA profile, it says that in column 2, allele obtained from the underwear of the victim, exhibit mark this, those obtained from vaginal slide of are mentioned respectively in columns 2 and 3. Column 4, which deals with pubic hair of the victim, reveals that there was uninterpretable low STR DNA autosomal profile was recovered. In the both cases, sir, in the Y chromosome also, in the autosomal also, they did not get proper profile, it was uninterpretable. Now in last column, profile obtained from the blood of accused is given. Thereafter 3 points are given, which are roughly translated as, from the source of underwear, this vaginal slide, autosomal STR DNA profile is obtained. Both the articles are pertaining to the victim. Thereafter it is mentioned that uninterpretable low autosomal STR DNA profile is recovered from the pubic hair of the victim. In the third row, it is mentioned that alleles obtained through autosomal STR DNA profile from the each genetic marker found on underwear, vaginal slide of the victim reveals that the alleles obtained, though autosomal STR DNA profile from the blood sample of accused, are not present on all the alleles. Actually, court has written in its own language, sir. I have just copied down from that order sheet. Actually, obviously, sir, in Madhya Pradesh, the reports are given in Hindi. It is being generated in Hindi. First point of discrepancy is that, now court observation that has, sir, is that exhibit P38 dated this in relation to accused, which was dispatched by Joint Director Regional FSL Bhopal in favor of the SP Sagar. It is contained in only 3 pages. No attempt was made to obtain autosomal DNA STR profile in relation to accused, whereas autosomal DNA STR profile in relation to the victim is available, as is evident in exhibit this. Now in exhibit P38 on second page, Y chromosome STR DNA profile has been produced. Like second page of this P34, it is mentioned that uninterpretable low Y STR DNA profile was obtained from the underwear, vaginal slide, and the pubic hair of the victim. Reference is given to the date of this or which report P34 was forwarded by the concerned JD. Thus, it is evident that the figures for the victim as far as her underwear, vaginal slide and pubic hair were picked up from the previous report. Thereafter, markers obtained from the blood sample of accused have been mentioned. It is mentioned that it is not possible to match the Y chromosome STR DNA profile of accused with that of the victim in view of uninterpretable low Y chromosome STR DNA profile. However, testing was done, then learned scientists conveniently omitted to carry out autosomal STR DNA profile as was carried out in relation to the accused, as is evident from the figures given on internal page of this report P34 available on record. This is the first lapse. Second lapse is that once as submitted by scientists and also supported by director, that they had obtained low Y chromosome STR DNA profile while carrying out electrophoresis. And since no male DNA profile was obtained, therefore it was not possible to match it with that of the accused person. Then the finding that it is different from the autosomal STR DNA profile obtained from the accused is misconceived and conveys a wrong message. The thing is that the profile which was obtained was uninterpretable, but the language should have been like that the court could have understand. Thing is only this, sir. The crux of the entire this order of the court, the thing is only that it should have been presented as like that they could have understand. The main thing is the understanding only. So that is why I have mentioned this, just because of related to my topic, and it is also related to legal. I have got this in the theme. So that is why I have mentioned this. And even I open it for the everyone in the complete arena, that we must discuss some of the fact findings and the things, lapses and many such shortcomings related to these report writings, so that everyone, because everyone are reporting officers now, they can understand that what they should do and what they should not do. It is followed with that only, sir. Now, my next topic is need of recent advancements. As, sir, yesterday itself, many deliberations were there related to these topics. As Dr. Hirak sir has already mentioned about the inhibitors, about many things. So, recent advancements, we must think of it like, what should we use? If we have 3 commercial kits available, we are strict to that, and we are not thinking above it. We have many sophisticated instruments in our different divisions. We are not amalgamating it. The thing is that, like, sir, I went through many things when I got this topic. Like, we have LCMS. It can examine organic material. Why can't we go on that? So that we can discover the organic inhibitors. Because yesterday, Dr. Hirak sir was saying very true, sir, that these commercial kits have identified certain like 10, 8, 12, whatever inhibitors they have seen in their countries, in their areas. But it is not like for everyone. India is so diversified that here itself, in India itself, at many places you can find many things different, completely different. So we have the instrument. We must go on it. We must at least identify the organic inhibitors in that. We have ion chromatograph. We can go for that, with that for identifying the inorganic inhibitors. Like sir, Hirak sir was mentioning about metal inhibitors. Obviously, then we must go for that solution, the chemistry solution. We must find out the chemistry behind it. And these are the sophisticated instruments we already have in all laboratories. And fortunately, sir, we are thankful to Dr. S. K. sir. In this SMFC, we have got one LCMS also sanctioned. So this will help us, sir, greatly. And interpretation of complex mixtures. Sir, this thing is for the bench workers. They are really confused that is it due to contamination or it is a true allele. So all the senior officials, senior experts are here. Even I leave on everyone that how we should minimize these things, because we also have inducted some new assistant directors who are working in our DNA laboratories. So they will be benefited out of that, sir. Best possible methods should come to overcome alleles dropout, because this thing also hurdles a lot, sir. And then study of autosomal and sex-linked STR markers. Like in that case, okay, although it was not interpretable, but issues are there, sir. Many times, even I have seen that sometimes what happens, that YSTR kits which they have brought now are so sensitive that they find the amplicons and they easily amplify, and we get the profiles through YSTR. But in autosomal, we don't see the foreign or the male profiles. It is being masked, or it is not so well developed. So we have issues in that. So it is also, I'm leaving it for open discussion, sir. And the future aspects, obviously, sir, as Dr. Hirak sir has already given us a way that we must think on it, that what should be the future aspects for us. Like need indigenous studies. As he was mentioning yesterday, that he studied on it, inhibitors, and in fact, other things, techniques which he developed and got patent. So all scientists everywhere must think on it, that they should do some indigenous study. And as I have mentioned, we already have instruments. We can go for that. We can devise methods. See, what I have seen, that if we purchase a LCMS instrument, we have a 3 years warranty and we have application backup from them. But we always focus on the routine examination. So we are not extracting many things out of them. We need to extract many things out of them. Like, we can ask them to come many times, and we want to devise new methods. You please think on it. We will put some different samples on it, which is not related to this division, maybe. But we want to do that, devise a method. If we need to change column, please change the column. Likewise, sir, selection of other techniques to study the chemistry. See, the same thing which I had mentioned. So, sir, I am leaving all these things for the open discussion. Being a host, sir, I want to be benefited, and my people want to be benefited of your expertise and experiences. With those words, sir, I end my presentation. Thank you. Thank you, sir. Thank you.
Thank you, sir. Now I invite Mr. Raslan, who has a more than 10-year experience in forensic field and also has degree in law, in specializing in criminal investigation. Now he is working as forensic application specialist for Centol company with a special focus on collaboration in India.
Hello for everyone. Let me continue to introduce our company. So, modern forensic DNA analysis present expert with diverse challenges that demand fast, accurate, and reliable solutions. At Centol, our mission is to support forensic experts by delivering exhaustive tools and optimized workflows for every stage of DNA analysis. Let's take a closer look at the forensic identification of a person in DNA research. As a solution, we offer a full cycle of research, from the collection, storage, and isolation of DNA, from biological traces to analysis of data obtained. Any job begins with the preparation of workplace, because we all know perfectly well that a clean start a guarantee of best results. We bring to your attention a two-component solution for removing DNA from work surfaces. The reagent is ready for use, safe for human, and to reliably destroy nucleic acids. As the workload of the laboratory increases, our company's solution is automatic DNA extraction station Calibri. The principle of operation is based on the use of magnetic particles of the same series of MSOP. The advantages of our station are ability to scale depending of the needs of laboratory, which low loads and adapter one for 48 samples is used, which increasing loads. The station can be easily be converted to 96 well version in just 5 minutes. Saving on samples, simple, fast and friendly interface, easy for transportation with weight only 21 kilograms. The presence of UF lamp, isolation of DNA from different types of tissues with one protocol. All reagents using during the operation of the station are ready for use and do not require any manipulation, with the exception of Proteinase K, which is supplied in freeze-dry storage complete with a gelation buffer, which ensures that all components are stored at room temperature until the first use, after which the proteinase must be stored at minus 15-25 degrees. 96 version. The kit RealQuant H3 for DNA quantification and DT prime real-time PCR machine. The kit is based on the real-time polymerase chain reaction method using Tachman probes to increase detection sensitivity. The RealQuant H3 kit uses multi-copy loci of human genome. Amplification of multi-copy autosomal fragments different size allows a reliable assessment to quantity and quality of DNA in test samples and amplification of multi-copy fragments of Y chromosome to determine gender. The DT prime device, in our opinion, has a most convenient and friendly interface, an open system. The software also allows automatic calculation of required DNA concentration for PCR. Dilution must be 1 or 2 stage, if it's necessary. The next step that brings us closer to obtain information is the study of amplified products using a genetic analyzer. Our company also solves the problem from all the region necessary for the launch and maintenance of analyzer to the produce of genetic analyzer itself. I suggest you take a closer look at the advantages of Nano40506 analyzer, such as resistance to overload signal, sensitivity to low DNA concentration. So, our analyzer is ready for increase in number of dyes, which is, I believe, will naturally come, and our system will not lose its relevance for many, many more years. So next group slides will clearly show us dynamic range of analyzer. 15 nanograms in reaction, we see very low level of noises. 25 nanograms, 10 nanograms, 5 nanograms. And this slide shows us same level of sensitivity between Nano4 systems and 3500. The Human ID kit allows us to examine 31 loci, 26 of which are autosomal, and 5 loci that determine the sex of person, 4 of which in turn are aimed to establishing the presence of Y markers, which is very important in the study of cases of sexual crimes and the presence of mutation in amylogenin zone. On this slide, you can compare the numbers of loci and their significance to experts. The kit has a great identifier quality, which plays a key role in study of mixed traces. As you can see, the kit performs well in difficult cases and allows you reliably determine both minor and major components. The human ID kit also performs well when working with degraded samples, such as bones, where the degradation index can be more than 5 times. One of the key points of that which only one kit, it's possible to launch both conventional PCR with the stage of normalization DNA concentration and direct PCR reaction. If there are need for a more detailed study of Y markers, our solution is human Y. 34 Y estereoloci, 6 dye, high sensitivity, all these are key advantages of our kit. The logical end of DNA research is the interpretation of persistent genetic analyzer data. This stage is one of the most responsible and error-free. That's why it's so important to have reliable software. Synthol has developed its own software for human identification analysis, HAD fragment analysis. Key features of the HAD fragment analysis: compatibility with any kits and any instruments. So data from the data Nano 405 and 06 is analyzed without prior conversion and without any loss of resolution or quality. The only software on the market which is feature a simple and intuitive interface with flexible customization options. The ability to combine analysis across different panels, standards, and methods with a single project. Flexible, user-friendly editors for panels and standards. The ability to work with 8 dyes simultaneously. Advanced size standard search algorithm. Dynamic thresholds. The ability to forcibly adjust the matrix eliminates spectral calibration defects. The same mixed sample analysis from HAD fragment analysis software, GenMapper. As we can see, the level of noises is very low, and pictures has the same height of pipes. So, thank you for your attention.
थैंक यू मिस्टर असलान। सत्र को अब आगे बढ़ाते हैं और मैं मिस्टर अजय कुमार राणा से रिक्वेस्ट करती हूँ, साइंटिस्ट बी CFSL हैदराबाद, प्लीज आप अपना आकर ओरल प्रेजेंटेशन दें। और श्री AK शर्मा सर से रिक्वेस्ट करती हूँ कि इस सत्र में चेयरपर्सन मैं उन्हें बनाती हूँ।
Good morning, everyone. Respected
Hello, hello.
What I'm saying is they have presented everything and they are saying everything is right and something is wrong with others, right? And they will be saying, obviously they have come out to introduce in India. So, who are you? You are the end users. You are getting the software, you are getting the machines. Finally, you have to deal with it. So please clarify the things. Earlier, we are not having competitors. Okay, it was Thermo Fisher, and now Syntholi is coming, some else are coming. Yesterday also I said, no, if ask, definitely one question you ask, what advantages you have over the over the other machines that you are using for the last 20-30 years. Then you will be satisfied, right? So, please clarify anything. You understood something or not? Why am I getting doubt कि आप सुबह के सो गए क्या? Because they are speaking something, are you understanding everything or not? When I come to a conference, try to grasp something. This opportunity will not come again and again. Right? And always I tell you, forensic scientists, the job of forensic scientists is to think. If you are not thinking, you are not going to progress. Right?
Good morning, all of you. I'm Ashwant. Since you have compared advantages and disadvantages across all the DNA analyzers present over there, one of the constraints in this day and age is the cost per sample. So can you please clarify as to how efficient your system is compared to others on cost per sample basis? If there is anything related to this, please let us know. So the cost per sample is what I am asking about.
So, about price and cost of analysis, it's much more less than a certified zero-zero system, first of all. When you use our system and our kits, you use only one kit for amplification, and then genetic analysis starts. When you use other system, you need to start, first of all, two STR kits. For example, it's global filer and mini filer to get same number of loci, and then you must start 2 times your genetic analyzer. That's it. Increase your price of analysis. And in future, certified 0-0 system has only 6 die versions, but in future, I believe it will be increased until 7 or 8 version kits. That's why you must have ability systems to these samples, to these kits. Thank you.
My question is to Ruslan, that excluding Russia, how many installations of their genetic analyzer are in Europe as well as in Asia? Approximate number. In Europe, excluding Russia.
It's around 10%.
10% of what? Total genetic analyzers. And in Asia? In Asia? Huh? Asia I'm asking.
In Asia, we have genetic analysis in Vietnam, in Vietnam.
Hello, good morning. Is there any case that solved by that Synthol kit which has been failed by other kits right now we are using like Thermofisher and Kyagens? I mean as than the sensitivity of the kit.
We have such big pictures, but in our presentation doesn't include it because it's a different time of use, and now we have enough of time to prepare it. But we have some results when, for example, verifier kits it don't show full profile, but our kits it show it.
Okay, thank you.
So we can start with the session. So we have to complete the session till 11:15.
Can I make a last question, sir?
Yeah, please.
Sir, we are satisfied with the consumables, Mr. Scientist. No problem. Your kit and other consumables. But see, you are comparing with 3500. Now you must be aware that next, what is that? Studio Flex, right? Studio Flex has already come, right, into the market. So comparatively, how better you are, your Nano 4, 5 or 6 than Studio Flex system? Because
yes,
they must be having advanced analysis software also. That you are telling your is HID FP, right? Your software. So now in Studio Flex, they will not be using IDX. They will be using next version of software. So that comparative study actually we wanted.
So I'm sure that GenMapper users will not have any problems to use our software because it's have same design. In our table, I can show for everyone if you want our software for HID kit, analysis kit, and here our software have same report system, same dye collection software. That's why it's not problem. And about sensitivity of systems, it have same sensitivity and same level of interpretation of raw data analysis.
Thank you very much. So we can begin this session. I think there are 4 presentations. So each participant will get 5 minutes, and then there will be a discussion for about 1 minute, right? So we'll be able to finish it within 11:15 or 11:20, right? Okay. You start, Dr. Rana.
Good morning, everyone. Respected Director, Chief Forensic Scientist, S.K. Jain sir, DGP sir, Director, CFSL, Guwahati sir, Directors of all CFSL and FSL. I am Ajay Rana from CFSL Hyderabad, going to present on sibling identification using exhumed skeletal remains that we have performed in our laboratory. So the first case of exhumed bone analysis was carried out on the bone samples of Dr. Joseph Mengele. He was an anthropologist and he was known as the Angel of Death. He was from the Nazi regime and a friend of Adolf, and he used to do very cruel experiments on the innocent Jews. And after the World War II was over, he fled away from Germany to South America, where he was living around Brazil, Argentina, and Paraguay. And in 1979, he died, and after about 6 years, his body was exhumed, and it was analyzed in a very 6 or 7 years of time by none other than Sir Alex Jeffrey. But the relatives with whom his DNA profile was compared, they refused to take the skeleton remains, and now it is being used for exhibition purpose in Brazil. So what are the basis of human variations? As we know that we have 23 pairs of chromosomes, and at each pair, one comes from the maternal side, the other from the paternal side. So the possible number of combinations we have is 2 to the power 23, that is 8.4 million combinations from a single parent. So the zygote is having about 8.4 into 8.4, that is 70 trillion combinations. Beside that, there are recombinations also, that is the crossing over during meiosis 1. And therefore, each individual on this planet is unique except monozygotic twins. So when we receive forensic samples in our laboratory, we try to compare it with the direct relatives, those who give the direct alleles. So we analyze either with the parents or the offsprings. But when these are not available, then we have to go for the collateral relatives. And they may be the brothers or sisters, and they may be first degree, second degree, or higher degree of relatives. So these are the collateral relatives. So collateral relatives do not directly inherit the alleles, but they share among them the allele. So they may share 2 alleles, 1 allele, or 0 allele. And according to that, based on some identity by descent, Hardy-Weinberg equilibrium, and Bayesian probabilities, this formula has been devised by Charles Bremer, and you can calculate the likelihood ratio of calculating the full sibling or half sibling. So when we obtain the sibling index, we have to interpret it. So if it is more than 1, then it supports the relatedness. If it is less than 1, then it supports the towards the unrelatedness. And if it is 1, then it is inconclusive. So the weight of the evidence is very low here. So this is the case we have performed in the laboratory here. The 2 terrorists were killed in Kashmir region, and in 2004 their body were buried, but dispute came into existence that 2 families, separate families, claimed that they were their relatives. And the case got lingered for about 20 years, and in 2022 their body remains were exhumed, and along with the alleged claimants, the bone samples and the blood samples of the alleged relatives were sent to CFSL Hyderabad for analysis. So here in this our lab, we selected the compact portion of the large bone, the femur, and we had taken this for analysis rather than the cancellous part of the bone. So this is the condition of bone samples we received in the laboratory. As you can see, these were laden with the soil samples, soil as well as many contaminants, the bacterial, which are of course not visible. So first of all, we removed the soil sample with the help of the
Come to findings, final findings.
So we purified the DNA from the bone sample through organic method. Then these were the 2 families, and we got this sibling index of half sibling more than 1, 6.45, and it was matching with exhibit A1, in which the donor had contributed his blood. rest it was either excluded or inconclusive. So we compared the data obtained from two populations in India, from Telangana as well as from Madhya Pradesh, and the data were concordant. So we can go for higher increase of the weightage of the evidence through inclusion of the more markers. Here we had used just 23. We can go for 50 markers. Then we can go on analysis of XXTRs or mitochondrial DNA in case of the sister siblings, Y chromosome analysis in case of the male siblings, and we can also go on depositing the samples to some free database like MatchPro or Family Tree for higher degree of analysis up to 5 or 6 degree.
Thank you, Dr. Rana. Thank you. Any
you,
question, please?
Dr. Rana, my fundamental question is that,
Yes,
do
sir.
you perform the mitochondrial DNA analysis in this case?
No, sir. Sir, this is the case of the male sibling identification, and mitochondrial DNA pass from mother's side only.
Yeah. One more question from Raslan, sir. My question is that in case of exhumed bodies, is it possible where the DNA is in highly degraded form, is it possible to identify using HID fragment analysis?
So, first of all, you must clean up surfaces and then have enough of DNA from extracted DNA. That's why you must use such series of magnetic particles or other sorbents. That it's if DNA concentration is enough for first of all in short zone, you can receive
Suppose that
half profile.
in exhumed bodies where natural forces are there, sample is degraded, and we got the low profile DNA. So, is it possible to amplify in such a concentration that it could be positively identified?
I'm saying it's possible because DNA from bronze storage about 15 years we can do it, and I'm saying we must do more than only STR kits. Use Segner, for example, and mitochondrial DNA research.
Thank you.
Thank you, Dr. Rana.
Thank you, sir.
So next speaker is Dr. D. Srinivasan. His topic is When Evidence Speaks, Forensic Preservation of Fetal Samples in POCSO Case.
Good morning, respected chairpersons and guests and everyone who have gathered here. I am Dr. Srinivasan, currently doing junior residency in the Department of Forensic Medicine at AIIMS Patna. I am going to do a case report on forensic preservation of fetus samples in a POCSO case. First, this is my theme of presentation. We are going to study about the degradation and contamination of the biological material, then the preservation of the DNA evidence, that is the biological sample, and how documentation and chain of custody is maintained throughout the course. These are my contents. Then we'll go with the introduction. First, sexual offences cases are serious concern in India, especially for minor girls, which are dealt under POCSO Act. The case becomes more complicated if the girl becomes pregnant, and in case of advanced stage of pregnancy, the termination is also very difficult. In those cases, special permission will be required from the court, and expert medical opinion should also be considered. In those cases, in order to prove the paternity of the accused, we have to preserve the DNA evidence of the case, that is the main fetus sample. This case focuses how proper preservation of fetus samples, accurate DNA testing, and maintaining the chain of custody will be reliable to get reliable results. This is the case overview. First, a 17-year-old minor girl came to the police department having 24 weeks of pregnancy with history of sexual assault, and a POXO case has been registered by the police department. Then, during a high court hearing, the court considered the advanced gestational age and asked to make a medical board to Patna Medical College and Hospital to check for the feasibility of termination at this age. But the Patna Medical College Medical Board stated that MTP could not be done as per the MTP Act. But the court was dissatisfied by this opinion since it did not address the main problem. Hence, second medical opinion was constituted at AIIMS Patna regarding the feasibility of termination, and we gave a comprehensive opinion that at this stage the termination could be carried over, but in an that will be in an uneventful in ordinary course of nature. And one more thing to be noted is the pregnancy has, if the pregnancy continued for the victim, it would pose some significant psychosocial stress to the victim also. Hence, legal clearance has been granted from the court, and we proceeded with the termination. Then, coming to the main part, that is the forensic handling of the sample. The fetus sample, which was expelled, was thoroughly cleaned in a sterile manner, and a saturated solid solution has been made by sterile distilled water mixing with excess of salt, which is the main preservative to be used in case of preserving the fetus sample. Many of the budding doctors are still now in confusion whether to use formalin or salt solution. Formalin will cause destruction of the DNA into fragments, and also will act as inhibitor in the PCR reaction, which is the main mode of estimation of DNA. Then, coming to the blood sample, which is retrieved from the mother and the accused to check for paternity, the blood sample from the mother will act as a reference sample for the fetus. Then the fetus will act as for the accused. Then this is how the chain of custody is maintained. First, each sample is documented properly in a separate forms. The first form shows the blood sample of the victim, second one shows the accused, and third one is the fetus sample, and basic details of the case and for what purpose we are sending the sample for testing. Then here at the below we have to tick the what sample we are sending for estimation of the DNA. Then coming to the second page, here witnesses will sign in front of the police officer, that is the investigating officer will also sign. Nursing officer is the second witness, and the doctor himself is the third witness. After filling the form, and the samples will be handed over to the police, and it was submitted to FSL for DNA estimation, and the court ordered for carrying over the DNA estimation in FSL Patna, and the results came out to be positive. That is, the accused blood sample was matching with the fetus sample. Hence, this was a newspaper clipping after the judgment showing the headline as the unborn child has saved the mother's honor. So this is how the proper preservation of samples and maintaining the chain of custody helps the court in delivering the proper justice. Coming to discussion, I will be discussing some of the methods how the fetus sample, some of the samples will be preserved. For example, in case of biological tissue sample, it can be preserved in saturated salt solution, and especially for fetus sample, one more specialized preservative is there, that is DMSO, dimethyl sulfoxide. And in case of blood, it can be preserved in 4% EDTA vial, or we can use FTA cards, which is much more convenient in these days. And one more method for blood is we can collect the blood in dry gauze piece by soaking it in the blood, and it has to be dried at room temperature, and then it has to be packed sealed in a paper envelope to be transported to the FSL. Then the refrigeration should be maintained at each step. For example, if you are storing the sample at the custody itself, we have to keep it in a refrigerated state for in case of this dried gauze samples. In case of liquid samples or fetus samples, it has to be kept in a frozen, like freezing temperatures for about -20 to -38 degrees centigrade. These are the common preservatives and the method and collection package of some of the samples. And I will be briefing through the contamination and degradation part within 2 minutes. So in
Thank
contamination.
you, doctor. Thank you. Thank you. Any question, please?
Nice.
सभी को क्वेश्चन होगा शायद पीओसी इज द मोस्ट चैलेंजिंग थिंग्स आप सबको पता ही होगा सर डॉक्टर साहब मुझे क्लियर टर्म से बताइए कितने दिन प्रेगनेंसी के बाद आपको दिखता है फिटस एमटीपी जब करते हो यू कैन आइडेंटिफाई द फिटस आफ्टर हाउ मेनी वीक्स ऑफ प्रेगनेंसी
सर वी कैन आइडेंटिफाई आफ्टर 4 to 6 weeks
4 week के बाद you will be able to identify the fetus so why don't you separate the fetus and send separately आप पूरा बोतल में क्यों डालते हो?
In this case, it was more than 24.
नहीं, not this case. Common case I'm telling. Number 2. Saturated solution is ये सही है क्या? नहीं है सही सर. जब आप saturated solution DMSO पर बोलते हो ना सत्यानाश हो जाता है. Clear cut guidance है what you call Ministry of Health का. जस्ट ड्रेंच इट इन नॉर्मल सलाइन जो मिलता है जो इंट्रावेनस इंजेक्ट करते हो ठीक है तो इसमें ना कोर्ट बुला के सस्पेंस तक भी जा चुके हैं साइंटिस्ट क्यों निकला क्यों नहीं पीओसी से ठीक है और कोई डाउट पूछे इथेनॉल
or you can preserve it in ethanol that small piece of
Dr. Devinder, I want to recommend the ethanol because the ethanol quality of the hospital is very poor and it's full of inhibitors. So DNA profiling cannot be done anything preserved in the ethanol, right?
So just a small suggestion actually. We can also put few drops of antibiotic solutions because mostly for the degradation purpose it is the microorganisms which are responsible for the degradation. So when you are preserving tissue samples we can use antibiotic solutions so that the microbial decay can be prevented.
We can recommend this thing to the hospital so you can make send a circular.
So this is regarding the uniformity in the report, this thing. Just a small point I just want to add. Because we don't have any uniformity in the training of the expert.
Right.
Throughout the country.
Yes.
They are from different regions, they have different brains, they do accordingly, like as per their convenience. So, like this board, whatever sir is suggesting to Janser, it should draft something which people can be trained in a similar way so that ultimately they will give a report of similar or uniform this thing.
Yes, sir. Sir, I would like to add on this, sir, to my submission to Dr. S.K. Jain, sir. Sir, we already have SOPs for biological samples, but categorically, sir, we must go for the DNA samplings. Elaboratively, we should mention, we should adopt some new techniques and the things like, sir, Dr. Hirak, sir, he's mentioned that we must use that to avoid some decompositions on such kind of things. So definitely, sir, it is my submission, sir.
कभी कभी ऐसा भी होता है ना जैसे एमटीपी आता है उसमें सिर्फ मदर का सैम प्रोफाइल ही आता है बच्चे का भी नहीं आता है उसमें तो नहीं मिक्स होने तो ठीक है तो हम निकाल सकते हैं लेकिन सिर्फ सैंपल्स ऐसे ही आता है इसमें
We'll send both.
Thank you. Very good presentation of uniformity of reports and your presentation was also good. But now point is, at the DFS level, we have also prepared the standard operating procedures for analysis of DNA samples, and we have circulated to all labs, and it is available on our website also, number one. Regarding the equipments, we have also prepared the list of the standard equipments, and which is also available. And regarding the upgradation of the lists, we have constituted a committee also, And I think Dr. Hemjay is also one of the member of that committee, which is still pending, and it should be done very soon. And my point is this: it should be done by the next month, by number 1, number 3. And regarding Dr. Sharma, Dr. Devendra is telling, group, we are definitely going to create a scientific working group on different disciplines, not only biological sciences, but in chemical sciences, physical sciences, and digital sciences. That we have submitted to ministry. After the approval from the ministry, we will release it. And regarding the training aspects, and our 3 labs, the CFSL Pune, Bhopal, Guwahati, and CFSL Guwahati has been designated a center of excellence in biological sciences, and there will be academy for biological science only, and Pune for chemical sciences, and Bhopal for physical sciences. So this we have already decided. And regarding the digital, we have CFSL Hyderabad has been designated. So this is in the pipeline, and very soon you will get. And this is the first time the DFS is doing such type of a conference, where only thematic based conference is conducted to bring the all scientists of same discipline and same platform, so that we can discuss thoroughly and in detail, and it will be useful to each and every person. But my request to all the delegates, you should discuss and share your views with each other, and so that we can further improve it. It is more important. And this is our idea, and I think because technology is changing very fast, that's why we are bringing the different companies like Centol and Thermo Fisher, and what new is coming, and we will definitely implement. Thank you very much. In the next coming time, the forensic will be on the big height. Thank you.
Thank you, Dr. Jain, for your valuable inputs. Thank you, Doctor.
Thank you. Thank you, sir.
Next presentation is Dr. Dhirendra Singh Yadav. His topic is gene expression profiling in forensic body fluid identification, significance, advances and challenges.
Good morning, everyone. Honorable Chief Forensic Scientist Dr. S.K. Jain sir, DGP sir, Honorable Sharma sir, Honorable Mahapatra sir. I am Dr. Dhirendra Singh Yadav from CFSL Chandigarh. I will be speaking on gene expression profiling in forensic body fluid identification. It is one of the emerging and under advanced stage of developing for body fluid identification, which is one of the essential requirement and challenging, because we require not only identification, but we require its identification of the person, but we require the identification of the fluid also, that what fluid causes that resulted into that DNA profile. So why we require? Because it is critical in forensic evidence. One of the DNA can identify the person, but cannot tell from which tissue or which body fluid it originated. So we require to identify the origin of that DNA profile, because that is required to establish the events at that crime scene. And the conventional methods like chemical, enzymatic and immunological methods consume a good quantity of this biological fluid, which is already in very trace or limited amount, and sometimes it is very difficult to do both, that is the identifying body fluid and this body fluid as well as DNA profiling by the same trace evidence, because of their low specificity, sample consumption and false positive. So we go for DNA profiling directly. However, there is one advancement, like when we go for RNA extraction, we don't require separate sample. We can extract RNA as well as DNA from the same sample in the same protocol. Means starting from the same substrate of trace evidence, we can go for extracting RNA in a particular stage and DNA in next step. So, it is crucial in establish link already informed, and when we go for fluid identification by traditional methods, we require separate stains, and we assume like it is a semen, so when go for different tests. If it is blood, we go for different tests. If it is like saliva, we require separate samples for separate identification of body fluid. While it is in reality, we don't get such kind of amount so that we can perform all such samples, and sometimes we could not identify on like undergarments or something that the sample is saliva, semen, or any other fluid. So we go directly for DNA profiling. However, the question remains that from where they originated, and so we require this some non-destructive method, that is the mRNA profiling or miRNA profiling, which can give us additional information like which can identify body fluid, which can quantify DNA RNA degradation, which will help in knowing the time since death or the age of wounds or what causes determining cause of death. So ultimately, these methods in complementary with the DNA profiling like mRNA, miRNA, and DNA methylase patterns, if they are combined with the DNA profiling, we can profile uniquely from single trace evidence to all DNA profiling as well as individualization or identifying the stain. So, principle kya hai? This is, we have the same DNA in all molecules, all cells of our body, from hair to saliva or semen. Every cell have the same DNA sequence, but every tissue or every fluid have different mRNA expression, because our fluid tissue require different enzymes, different proteins, and that's why the expression of DNA, expression of genes is different from one organ to another, one tissue to another, one body fluid to another, and that's the principle that we can identify on the basis of mRNA transcripts which are present in these body strands. So that is the principle. The transcripts vary from fluid to fluid and organ to organ. We can identify which kind of cells are there, which are leading to this DNA profile. So ideal methods should include which can help in identifying type of fluid, time of deposition when it was happened, and individualization of the individual. So mRNA profiling will help in tissue identification, and it is compatible with DNA profiling. Means same trace evidence can be utilized for both identification of the stain as well as the. It is additional advantage. It is highly sensitive, highly specificity, and multiplexing ability, and it is compatible with DNA profiling also. So we can identify from the single kit whether it is blood, semen, saliva, vaginal fluid, or menstrual fluid, menstrual blood. It saves time as well as quantity. Already markers have been identified by different literature. It is available, and these markers are to be refined in a kit, single kit, which can provide proper, which can provide a appropriate multiplex conditions so that it can be utilized in cases of forensic, actual cases of forensics. So challenges mRNA stability. It is unstable molecule. However, it is when we are going for miRNA and other samples, it can be utilized as a very useful ideal solution for the identification of stains as well as for DNA profiling. Even SNPs can be done by the same. So advantage of mRNA profiling is that it can be recovered, and it can be integrated with miRNA profiling as well as stability markers and multiplex panels can be improved. And complex analysis can be helped with the help of AI. So mRNA profiling enables body fluid identification, individualization, and may provide more accuracy about time since death. And it is highly useful in complex and mixed samples. So DNA profiling, when integrated with mRNA and miRNA profiling and epigenetic approaches, are emerging as next generation forensic standard, which can further revolutionize the field of forensics.
Thank you, doctor.
Thank you very much.
Any questions, please?
Very good morning, sir. Sir, my knowledge about mRNA expression is that it is not specific. It varies as per the need of the body and the need of the organ. So not all the mRNAs or genes are getting expressed at all the times. So I want to understand the underlying principle as to what are we detecting? Like if we are detecting the expressions of mRNA, there are specific mRNAs that we are detecting, or is it a generic concept applied across the various biological samples?
ये mRNA जो है यूनिफॉर्मली एक्सप्रेस नहीं है, वो मैं पहले ही स्लाइड में बता चुका. इट इज DNA is uniformly present in all cells, but mRNA quantity as well as quality, which kind of mRNA, which kind of genes are expressing a particular strain, is decided by the requirement of that fluid. Like I have also written in this slide that like they are creating a particular जो genes हैं, they are required in specific organ only, specific cells only. That's why they are expressed in there and remaining silent in other tissue.
Thank you.
Okay. जिस बॉडी पार्ट में जिस जीन का रिक्वायरमेंट है वहीं वो एक्सप्रेस करता है, और जिसमें उसकी रिक्वायरमेंट नहीं है वहां पर वो नहीं कर रहा है. अब कुछ सेल्स, कुछ जीन्स ऐसे हैं जिसमें एक से ज्यादा ऑर्गन्स में वो एक्सप्रेस कर रहे हैं, लेकिन हमने कौन से मार्कर्स आइडेंटिफाई किए जो पर्टिकुलरली उसी टिश्यू में, जैसे सीमेन में ही एक्सप्रेस कर रहा हो या केवल वेजनल उसमें एक्सप्रेस कर रहा हो लेकिन और में नहीं, उनको आइडेंटिफाई किया गया. Actually, my PhD work also involved gene expression profiling by mRNA profiling at ICMR Delhi.
Next question.
Can any case findings present in which mRNA identifies the origin, or it clearly differentiate either this profile is coming from the epithelial cells or other body fluids?
Oh, your actual case, it is still in research. It is a part of research.
Do you know there is no any finding has coming out from any cases in which the clear-cut profile is present, either it is coming from the epithelial cells or other body fluids?
They are doing, and they are combining. This is part of research. Still no kits are available as on date, but they are working.
As you told, it is from mRNA identifiers. mRNA identifies the origin of the fluid, but
yes.
Till now it is in a not any single finding has been coming out from any.
No,
type of
there are
research.
panels are there. Panels for there are several, like the markers which I mentioned, like two markers. Other than two, there are several list of markers. But requirement is that these all markers should be combined in one kit to make it useful for forensic purpose.
Thank you.
So,
whatever it is, हमें खर्चा नहीं होता है, 50 रुपये भी खर्चा नहीं होता है. In this context, हम इसका किट यूज क्यों करेंगे?
सर कंफर्म टेस्ट करते हैं वो.
अगर हम डिटेक्शन करेंगे, सीमेन डिटेक्शन करेंगे, सलाइवा करेंगे, मिल्क करेंगे, व्हाटएवर इट इज, वी कैन डू गो फॉर कंफर्म बेंजीडिन टेस्ट करो, देन टाकामा टेस्ट करो, देन एपी टेस्ट करो. इसमें खर्चा भी नहीं होता है. Not laboratories. So who is going to spend this much purchasing specialized kits, specialized machines, specialized multiplexing these things? हम क्यों करें?
सर शुरुआत में जब सैंपल 90 more than 95% cases में we have sufficient sample, but there are samples, there are cases where we don't have enough sample to identify by traditional methods. If we use traditional methods for identification of fluid, it will destroy, it will consume that sample. I am telling about actual case of DEVU, review case where they have all already cut several marks in undergarment of a victim, and that was used in serology and biology examination. They used in this DNA examination, and that state came to be negative. And when I found very minute stain and I profiled a positive DNA, but if I utilize that sample for identification of the fluid and type of fluid.
Got it, Jadhav sir. But to the best of my knowledge, sensitivity of spectroscopic analysis is 50 lakh, I think 50 lakh or 5 lakh something. Thank you.
Sir, thank you. Thank you, sir. Thank you, Doctor. I'm grateful to all the speakers, and they have nicely replied all the questions. I'm also thankful to Dr. Raslan for giving a nice presentation. So hopefully we are coming to end of this session and we are going into a tea break. But before that, I would like to say I have seen few presentations yesterday. The victim sexual assault's victim's dignity should be maintained and it should not be disclosed. Some of the slides had the media cuttings or where the name was mentioned. Media can defend itself, but we are the government servants. No one is going to save us, right? Being a responsible government officer, it's our duty that sexual assault victims' dignity should not be disclosed at all. Thank you very much. We are breaking for tea session, and after 15 minutes we'll return here. Thank you.
अच्छा तो मतलब ये ठीक ठीक ओके 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 ठीक टी ब्रेक के बाद पुनः आप सबों का इस सभागार में स्वागत है। हमारे चेयरपर्सन बहुत जल्द ही आ रहे हैं और प्रीवियस सेशन हमारा कंटिन्यू रहेगा। जब तक हमारे चेयरपर्सन नहीं आ जाते हैं तब तक इस एस्पेक्ट्स में अपना एक घटना आपके साथ शेयर करना चाहूँगा। ये घटना 2021 की है। सर हमारे चेयरपर्सन पहुंच चुके हैं। इस सत्र के जूरी मेंबर के लिए मैं श्री दिलीप सर, डायरेक्टर एफएसएल भागलपुर को सादर आमंत्रित करता हूं। हमारे दूसरे जूरी मेंबर हैं डॉक्टर कमल चौहान, सीएफएसएल दिल्ली को भी मैं सादर मंच पर आमंत्रित करता हूं। अपने इन अतिथियों के स्वागत हेतु मैं आयुष कुमार से आग्रह करता हूं कि वो पौधा देकर दिलीप सर को स्वागत करें। अब मैं सुश्री सीमा पटेल, सहायक निदेशक पटना से आग्रह करूंगा कि वो पौधा देकर हमारे जूरी मेंबर डॉक्टर कमल चौहान का स्वागत करें। डॉक्टर A.K. शर्मा सर से अनुरोध है कि वो अपने सत्र को जारी करें।
So next presentation is by Ms. Madonna Matthew from Usha Martin University, Jharkhand. Topic is environmentally modulated and arthropod succession and decomposition dynamics. Digitally assisted, serial number 15, assisted forensic entomology observation study from Angara, Jharkhand. Apologize, the topic is different. Forensic applicability of endophytic signature from Datura stramonium in Angara, Jharkhand. Implications for trace evidence and PM analysis.
Hidden truth often lie in the most unexpected places, even within toxic plant. Science find evidence that speak louder than words. Today, we are going to take you into fascinating world where plants don't just grow, they actually help in solve crimes. Good morning. I am Deepika Kisku.
I am Madonna Matthew.
And we are here to present our presentation on the topic forensic applicability of endophytic signature from Datura stramonium. This is the content. Datura stramonium is also called Jimsword, or it is a toxic nightshade plant rich in tropin alkaloid that can cause hallucination, delirium, and even death. Beyond its toxic, it carries microbial fingerprint that can serve as powerful, eventually tools in poisoning investigation. Sorry for the interruption. Plant-derived toxic poses serious risk in both accidental and intentional crime, making rapid investigation critical in forensic science. Endophyte microbes communities act as biological fingerprint, enabling precise source tracking and strengthening post-mortem interpretation.
So moving on to the methodology, samples of Datura stramonium were collected from our location Angada, Jharkhand. Then the surface sterilization was done using running water. Sodium hypochlorite was used as a sterilizing agent, distilled water and ethanol. Then plants were placed in the Na and Pedia medias for the growth of fungus and bacteria. And as per our results from 6 different parts of Datura stramonium, stem, root, fruit, seeds, leaves and flowers, we have obtained a total of 16 isolates of endophytes, 7 bacterial and 7 fungal, 9 bacterial and 7 fungal ones, which shows good results with the pH tests and the temperature, and which gives positive results to the metabolites, and different tests for alkaloid, flavonoids, tannins, etc. were done. So microbial traits were consistently linked to the Datura stramonium. This is again a region-specific study, because we want to state that even though the plant we have taken, Datura stramonium, comes under toxic plants, and there are many cases in Jharkhand related to accidental poisoning and mass animal cattle deaths as well. So, and it's widely available also, but still, even though every part of Datura has toxins in it, as we mentioned earlier, hyoscyamin, scopolamine and atropine, but after all, even though in every part will have specific endophytes, both bacterial and fungal, which can be beneficial for future uses in forensics as well. So endophytic communities acts like natural fingerprints identifiers. Alkanoids, flavonoids and tannins detected in isolates serves as biochemical markers. Distinct bacterial and fungal morphologies were found and categorized. So this adds a robust biological layer, enhancing admissibility and credibility to forensic evidence. So I'll ask Deepika to continue with the future direction.
Development of region-specific forensic database, training forensic professionals in microbial evidence analysis by combining microbial fingerprint with metabolic and enzyme profile, decoding death. After death, plant toxic interact with microbial communities, altering decomposition, dynamic and shaping microbial succession. This microbial shift provide tempered clues that add the depth to traditional forensic methods, supporting post-mortem interval. The forensic applications are endophytic signature, strengthened trends evidence, linking and connecting plant fragment to their source after transferring and decomposition. They support poisoning investigation, post-mortem interval estimation and courtroom administrability by offering the biological layer that affect the environmental noise. So here I conclude that the Datura stramonium rapidly identify the plant poison in crucial for both medical treatment and forensic investigation by integrating microbial fingerprint into forensic workflow. We enhance trends evidence linking with the cases and legal proceeding while raising awareness of hidden societal risk. Thank you.
These are the references, and we are open to questions.
Questions, please.
Okay, thank you so much. Yes, sir.
So, you just in your result you showed that Bacillus and Aspergillus is the
Yes.
community signatures. They are the most ubiquitous microflora. So how can they be the microbial signatures for the identification of these plant species?
Yes, sir. Thank you so much for the question, sir. This is a part of my PhD thesis. I'm currently working on it. This is my second year only. So Dhatura is the plant I have taken, and also Atropa belladonna, because both have the same toxin atropine, and that is mostly there. So till now, only I have got 16 endophytes, but there are more to see. But as of now, we have only concluded Bacillus and Aspergillus. But yeah, we will be focusing on the least, available. Thank you, sir.
Again, just a small suggestion. If
Yes,
you are,
yes.
Don't go for only genus level of identification. Go up
Mon-
to species
mon-
level
yes,
of
yes,
identification.
sir. Thank you so much.
So next speaker is Ms. Deepika Sharma, again from the same university. Integrated forensic analysis of arthropod succession and decomposition dynamics in Angara. So I think this is a similar topic. Using ecological indices, physicochemical profiling, and digital taxonomic validation. I call Mr. Vedant Biswas. His topic is integrating forensic anthropology, odontology, and DNA in disaster victim identification, strengthening forensic. Again from the same university.
Good morning, sir. Good morning, everyone. I'm Vedant Viswas.
I am Pritam Kumar.
And we are the students of BSc Forensic Science, Usha Martin University from Jharkhand, Ranchi. And our topic is on integrating forensic anthropology, odontology, and DNA in disaster victim identification. These are the contents. DVI, it is a forensic process that used to identify victims of mass disaster like natural calamities and accidents or any kind of terrorist attacks. In these days, DVI is becoming very more organized and scientific, that it's been helping experts to identify and manage the victims using better methods and technologies. It contains forensic odontology, DNA profiling, fingerprints, radiology, and digital data for more accuracy of decomposed and burnt cases. In 2026, DVI is not only working, forensic is not only focused on identifying victims, but also treats the victim with very respect and dignity, and also helps the victim's family. Why DVI is very much important in India, why we need it, because situations like floods, earthquakes, crash, and industrial accidents really happen often. So having a nationwide DVI is really important in 2026. Also, sometimes it's hard to identify the victim because of badly damaged body and missing records. These are some examples of real life incidents. First, we have Indian Ocean Tsunami 2004. It happens in huge scale, geographical dispersed death, and very poor documentation. Second, we have Uttarakhand floods 2013, 6,000 deaths and misidentification until DNA profiling really happened. And then Mangalore air crash 2010, mostly charred body and same misidentification until DNA profiling is done. Kozhikode air crash. It wasn't that much big, but still 21 fatalities, and really slow identification. Balasore train crash. It's mixing of Balasore train crash 2023. Mixing of remains, and it exposed the India's weak identification infrastructure. These are some role of DVI forensics. Role of anthropology, it's basically focus on biological profile, age, sex, statures, and ancestries. Then role of odontology, it totally depends on dental records for the identification, useful in mostly decomposed and burnt cases. Role of DNA, it is the most accurate method to identify victims, only used when other methods were failed, and it requires proper data for the identification. Thank you. This is only from my side, and rest of the slides will be taken over from Patel.
Proposed DVI
Any
framework.
questions, please? First time I am seeing two speakers of the same topic. Is it allowed? I won't allow. Please get down. I won't allow.
Thank you, sir.
Next presentation is by Ms. Annie. High performance TLC for the detection of anti-tumor compound Vidafrin A in Soxalate extracted crude extract of ashwagandha root. I don't know why this topic is in forensic biological sciences. I don't know. So next is the last presentation today is Mr. Anas Rabbani.
A very warm good morning to all the members present here. Myself, Annie, research scholar from Patliputra University, under the supervision of Dr. Munawar Fazal, Professor, Department of Botany, College of Commerce, Arts and Science. So, today I will be presenting a part of my research work. I will begin by sharing the outline of my presentation, which include the key section I will cover. So I start with the topic first. The topic is a high performance thin layer chromatography for the detection of an anti-tumor compound in Soxalate extracted crude extract of ashwagandha root. So why I have selected this topic? What is the reason behind that? Actually, I realized that people in the rural area often struggle to afford or access the cancer treatment. This inspired me to explore how we can use natural resources to develop low-cost anti-cancer medicines. I chose this topic because I want to bridge the gap between nature and the medicine to help those who need it the most. So for that, I have selected one plant, and the name of plant is ashwagandha. So I start my work with dividing my steps in 4 steps. These are, first one is the crude sample preparation, second one is the high performance thin layer chromatography, And third one is the breast cancer cell line. Fourth one, the MTT-based cytotoxicity assay. So for the first objective, I have taken 50 gram of root powder with Aeneas somnifera, added 500 ml of 80% methanol. After Soxate extraction, I get a paste. Then I perform HPTLC. For that, I have taken a stationary phase, and the stationary phase is silica gel 60F to 5 plate, and dimension is 100 into 100 mm. And for the sample application, 8 mm from the bottom of the plate is required, and the solvent font I got is 70 mm. The model I have used for HPTLC is Linomet 5, TLC Scanner 4, Visualizer 2. And this is the track assignment for the qualitative determination of the test compound. So for that, I have taken 3 lane, 2 lane contain sample and 1 lane contain the standard. And for that all, I have taken 1 mul sample. Then I make the crude calibration curve. For that, I have made different concentration of the standard, and then it is plotted against the peak area that I get after the HPTLC. And these are quantification of the standard and sample. After that, I culture the 2 breast cancer cell line, MCF7 and MDMB231 cell line on the DMEM media. After trypsinization, the cell get detached and observe under the inverted phase contrast microscope. This is the principle of the MTT assay. Actually, MTT is a yellow color dye. When it combines with the live cancer cell, it forms a formazone compound, and it has purple color. It gives purple color in the spectrophotometer. So intensity of this is purple color. We have to measure by using the spectrophotometer, and these are results. This is the powder that I have made from the Sugandha, and these are paste after Soxate extraction. And for the qualitative determination, total 3 track I have used, and we can see here, in all the 3 track, band has come at 0.4 RF. It indicates clearly that yes, this extract contains the anti-tumor compound, that is vitafurin A. For the quantification, I have run the sample 9 times and sample standard 3 times, and it matches at the lane first, and this lane first contains 0.5 μg per ml. This is the result of MTT assay, and here we can see in control, the all cells are live, so 100% cell viability, and at the higher concentration, the cell viability is less. These are some morphology photos that I have got after the inverted phase contrast microscope, and in control, the cells are all very much healthy, and they are uniformly, but at higher concentration of this extract, the cell get clumped with each other, and they are glowing in nature. It means yes, this compound act against the breast cancer cell line. And in conclusion, I must say yes, this extract contains the anti-tumor activity, anti-tumor compound, and it works against the breast cancer cell line. And in last, I would like a big thanks to the Vice Chancellor of Patliputra University, my supervisor, Dr. Dimple Ma'am, Coordinator Biochemistry Department, the Organizing Committee of Setcon 2026, and family. And at last, I want to conclude my presentation here. Thank you. Yes, sir.
You extracted Bithania somnifera powder, and thereafter you identified that one anti-tumor compound in it was able to suppress the cancerous cells.
Yes, yes.
So have you you have compared it with the control?
Yes, yes.
Have you conducted any study? Have you conducted any study to identify that compound which was showing RF 0.5?
Yes.
Like mass spectroscopic study?
Yes, a GCMS I have done, and in GCMS it is confirmed that yes, it comes on the 0.4 RF. Okay. And here also one slide I have shown here, it is coming on the 0.4 RF only. See here, the all band are coinciding at 0.4 RF only.
Okay.
My slide is not visible.
So this result of the anti-tumor compound was comparable to the GCMS.
Yes, GCMS
results
also. Yes, I performed GCMS also, and this is the result of HPTLC.
I know.
Yes.
This should be confirmed with the GCMS.
I confirmed already, sir, GCMS.
Thank you.
Thank you.
एक छोटा सा अनुरोध है कि जिन पार्टिसिपेंट्स का ओरल प्रेजेंटेशन अभी तक नहीं हो पाया है, कृपया वो अपना नाम हमारे वॉलंटियर मिस्टर आलिम को दे दें। लंच ब्रेक के बाद उनका ओरल प्रेजेंटेशन होगा। धन्यवाद।
Next speaker is Mr. Anas Rabbani. His topic is application of artificial intelligence in wildlife forensics, a conceptual framework for detecting poaching and trafficking in eastern India. From Usha Martin University, Jharkhand.
Good afternoon, everyone, all the participants and jury members. So, my name is Mohammad Anas Rabbani. I'm from Usha Martin University. I'm a second-year student of Bachelor of Forensic Science. Today, my topic is application of artificial intelligence in wildlife forensics. So, without further delay, let's move. Introduction to wildlife crime. Wildlife crime refers to illegal activities against animals and plants. Includes actions like illegal hunting, poaching, and trafficking of protected species. We all know there are several acts regarding Wildlife Protection Act 1972. India has a rich biodiversity with about 8% of the world's species, which make it more vulnerable to wildlife crime. Now moving the situation of wildlife crime in India, because we are focusing in India right now. According to recent data, more than 2,700 wildlife crime cases were reported between 2020 to 2024. In 2020 alone, 820 wildlife crime cases were reported, the highest in recent years. Also, 30 to 40% of wildlife crime incidents may go unreported. Wildlife crime in India is not only limited in poaching, but also includes in illegal trade, animal parts, smuggling, and habitat destruction. As highlighted in these points, you can see. Let's move to the next slide. These are the challenges in wildlife crime investigation. So, as we know, wildlife crime investigations, the major challenges are to species identification. For example, like the investigators found some bones, scales, or skin, or anything about like wildlife. So how will they identify it? Because we do not have a particular or a developed research facility or trained experts to identify the particular wildlife species or any of that. And also, India has a very large of rural areas and remote areas. So how will they identify it, and how will they report it to the particular organizations, or how they will identify it? So this is the map. This is a core part of the PPT on the presentation. As you can see, these are the trafficking routes around the eastern India. Mainly these animals like tiger, pangolins, and snakes, snake venoms, they like poaching and hunting, and for medicine and for other purposes, these are various days around the middlemen through the forest, because they cross the international borders of Myanmar, Bhutan, and other areas. So we have the main role of artificial intelligence in wildlife forensics. Automated species identify using machine learning and image recognition. Although many countries use these methods like camera trapping and using tiger with the collars used to track them and where they're going around. So like many countries like Kenya or South Africa use these methods. Drone-based surveillance for monitoring remote forest regions. Mainly like areas like Madagascar or in around Argentine, people use like the monitoring remote forest regions in Brazil and in California districts. Mainly our vision is to propose an AI-based forensic framework. As you know, AI is increasing in the world, and also in India, AI is everywhere. AI is in mobile, AI is everywhere around the world. So our vision is to mainly these are the visions of our, to mainly encourage and to solve wildlife crime preventations. And so I will conclude the topic. Wildlife crime poses a serious threat to biodiversity. As we know, know that the wildlife crime is a very big trap networks and all the large areas vast in the crime scenes. Eastern India is vulnerable due to rich biodiversity and cross-border trafficking routes. Traditional investigation method face significant challenges. Artificial challenge can strengthen wildlife forensics investigations through improved monitoring, species identification, data analysis. Thank you.
Any questions, please? So we are ending this session here.
Thank you.
And we are going to have the poster evaluation, and the jury member for the poster evaluation are Dr. B.K. Mahapatra, Director CFCL Delhi, Dr. Devinder Singh Yadav, CFCL Chandigarh, Dr. Devinder Kumar, Director FSL Andaman Nicobar, and Shri Himjay Kumar, Director in charge, Patna. So, thank you very much for all the presentation you had and all the participants and the delegates who have interacted with the speakers. Thank you very much.
Thank you so much, sir. And as a poster evaluation is going to start, so I request to everyone and the jury members, please go and check the posters in the gallery and evaluate it. If you have any questions to ask who is going to present their posters. And before going to the poster evaluation, I would like to thank my chairperson and the jury members by giving them memento. So I call them Srimati Shalini, Sushri Garima Singh, and Shri Saurabh Suman. One more announcement is there. So during the poster evaluation, I would like to tell Mr. Eager, prepare your lecture after the poster evaluation, so it will be continued. Thank you. So you may proceed for the poster evaluation with the jury members. Thank you.
Sorry for interruption. Announcement is like this, that during the evaluation of poster, Mr. Iger will deliberate on his topic so that the rest audiences will have the time to at least sit and look after his deliberations. Thank you.
presentation, sir. It's date modified. Date modified? No. No. Date modified. And pick once again, please. It is the right, sir? This one? No. The destination? This one. This one.
So this session we have decided will be chaired by Hirak Ranjan Das and his colleague Mrs. Dr. Sama. And I request जिसका presentation है poster presentation, so please come to their poster side. They will be explaining the things, they will be describing things, and rest of people will be here only to continue the lecture. Thank you. Can I have sufficient time, sir? Explain clearly and slowly. Right? Okay. Thank you.
Thank you, Dr. Mahapadra, to make me a chance to explain more about our new genetic analyzer, Nano 406, which you can see on this picture. And the main feature of this device is that you can work with three types of different capillary arrays. And depending on the throughput of your laboratory, if you do everything every day, more than 100 samples, you can use 24 capillaries. If somehow the number of samples less, you can change this array to 8 capillaries, and you can use the same machine. Another important feature, you can see Nano 405 and Nano 406, and it's smaller, it's a more compact device. When you apply the
Форезы дверь открыть невозможно.
In the old machine, if you open the doors during electrophoresis stopped, your analysis broken. So in this case, no problem. The door is blocked. Okay. It also has 2 plates, so you can put inside this device 192 samples, 2 plates. According to the data, here is the picture of allelic ladder from Nano 406. And this is data from samples, general samples we analyzed in St. Petersburg, in Russia. You can see here the clean STR profiles for positive sample. And because Dr. Mahapadra asked to compare Nano 406 with other machines, we prepare some slides. It's from the left side, it's a ladder from Nano 4, and from the right, the competitor YA. This is positive control. This one, 50 nanograms DNA per PCR, which is large quantity, but you can see from the left side is very clean STR profile. 5 nanograms as well, and 0.31 nanograms also the same sensitivity. And let me show you one interesting example of our experience to work with ancient DNA. In Crimea, which is on the south of Russia, we have the ancient city artesian. And last year, archaeologists found the remains of warrior. According to radiocarbon analysis, it's about 1,060 years old. So they asked us, do you answer who this warrior? Is this Caucasus or Tatar? Because at that time in Crimea, it was the border of times between Sarmats and between Tatars. So we asked to provide us 2 samples. They provided us by tooth and a small bone from the finger. And we extracted DNA, and using real-time PCR, we found that very, very small quantity of DNA, very low concentration, as you can see, only 10 picograms per microliter, and we have only 50 microliters of this sample. And another point that this DNA was, of course, very degraded, so you can see long DNA fragment has even less quantity, about 1 picogram per microliter. And another thing that this warrior was a man, because we found Y chromosome. So first of all, we have done STR analysis to be sure that no contamination in these 2 samples, these 2 DNA samples. We used 2 different kits, and we found that everything is okay, very clean, but very degraded profile, as you can see. Next step, we analyze Y chromosome. We used the kit for 34 allelic loci, but found only 14 because of high degradation. That was not enough to understand what kind of man this warrior, Sarmat or Tatar. So we start mitochondrial DNA sequencing. And you can see here the sequence from DNA man who died 1,600 years ago. It looks like very fresh, very modern. And when only 2 microliters was in the tube, we started to think how to prove whether this man Sarmat or Tatar. And Professor Kornienko supposed, let's say SNP, single nucleotide polymorphism analysis for haplogroup J2 M172, which is Tatar of Crimea, and we have positive result. So we provide archaeologists the information about this man, and after that they sent this sample to United States, and they used the special test for 1 million SNP analysis, and that test proved our result. But our result we got for very, very small money and very fast, for maybe 1 week or maybe 2 weeks. So that's how this technology, the simple technology, real-time fragment analysis, Sanger sequencing, help us to work with very ancient, very complicated samples. At this slide, you can see our main features to work together. This system we are elaborated, Nano405, Nano406, it's an open system. You can use every kit available for you. It's very robust. You know, this Nano4 here on the booth, it goes through 8 different places in India. It's 1,000 of kilometers, and it works very well, calibrated very well, so it's very robust and very robust not only for transportation, but high humidity, high temperature. It works very well. It has very high dynamic range, and as you can see, the high sensitivity. And also, we are ready to share with you with our scientific knowledge. If you have some difficult cases, some question, please ask. We have enough, many years experience to share with you, and also maybe you will share with us. And of course, we have very competitive price. So thank you for your attention. If you have any question, please.
Thank you, sir. Just I have one question, sir. Here, from here. It's me.
Okay.
It's me. Okay. So, does your Nano for 6 has both the modules, like genotyping and sequencing?
Both.
It has both?
Yes.
Okay. Then what are the kits that can be used for the Sanger sequencing purposes?
Sanger sequencing?
For Sanger sequencing purpose?
Yes.
It will be your kit, or it can be any kit that we can use.
Excuse me. No, it's open system. You can use big die, no problem. Big die, no problem. But we also have our own GenSec. It's like big die.
So when you are talking about open system, whether it is for with regard to kits or the consumables which are required by the instrument also, because for polymer, for ABC, CBC and everything, they need to be purchased from you people, right?
We have everything, the reagent, consumables, buffers, polymers, no problem. Our own production, we have very big production in Russia, so no problem.
So that means the polymers, ABC, CBC, that needs to be yours.
Yes, but you also can use another.
We can use any other one?
Yes, you can.
Okay.
Open for everything.
Open for everything. Okay, thank you. Any more questions?
Yes, please.
Yeah.
Your autosomal as well as the YSTR profile were the partial, right? And when we have the DNA which is highly degraded, it's very difficult to have the sequencing or the full sequencing. And did you perform the mini sequencing? What was the sequence size you did the sequence analysis?
For highly degraded DNA, we can sequence even 50 nucleotides because our kit starting sequencing the first base after primer.
Right.
So you can, in my previous report, I showed the case for 100 base pair sequencing with very high quality.
So basically mini sequencing. Right.
Mini sequencing.
Right. Okay.
Any other question?
Kit. With your STR kit, how many populations of European and Asian have been studied?
So your kit has been validated against which are the other populations?
Okay.
That's what his question is.
Okay. It's almost convenient for Russian population, of course, and Vietnam. But it's possible to add some loci special for other nation. because we are produced and elaborated these kits, we can easily to add 3, 5, 7 loci, no problem. And also we have the case and publication and the poster here about green boots. It's for Indian person identification. So it works.
Actually, sir, it's not about adding more STR markers actually. For at this point of time, the concern is that whether your allelic ladder, it contains all the alleles which are found in Indian population for a particular specific markers. But otherwise, in the laboratory, it will become really difficult to interpret an allele. Otherwise, that will fall in off ladder or OMR. Whether have you taken care of that aspect?
Exactly. You're right. Allelic ladder a little bit difficult to make, but if you have such a task, we can together find in population and combine new allelic ladder loci, no problem.
Any other question from student side?
Suppose we have a system 3500 of thermofisher. Can I use your consumable PAP, capillary, ABC, CBC, etc.? It will be compatible with 3500 technology we have?
Yes. On the booth here, you can see, I will show you the CBC, ABC, polymer, everything for this machine. We produce it.
And for your machine, their consumable is compatible? Suppose we have the Nano for 5.
Yes.
Okay, I have the POP for thermofisher, POP 4, okay. ABC, CBC of 3500. Can your Nano 4 is compatible with that?
Yes, compatible. Open system, compatible.
Open system.
Yeah.
Consumable wise and the STR. Oh, thank you.
Yep.
See, the main genetic analyzer supplier, they were not making the polymer. They were buying it from someone. Right? Let me name that MC Lab. Right? That was much cheaper in the earlier genetic analyzers. So we used to purchase that at 1/6 of the cost which Thermo Fisher was supplying here. Right? So the source may be different. Right? So you may be buying from the same source. Right?
No, we produce it by ourselves. Okay. Because it's 30 years experience. In Russia, we have a huge facility because 400 devices we installed and all of them work.
Just one more quick question from my side. Actually, whether your kits and consumables, you are validating them as per SWACDAM guidelines or you have any one set of guidelines of your own? Actually, mostly the US-based kits, they are being validated for forensic applications or forensic use as per the SWACDAM validation guidelines, SWACDAM guidelines, a scientific working group, SWACDAM guidelines. So your kits have been validated. So in that regard, your kits have been validated as per SWACDAM guidelines or any other guidelines?
Okay. Of course, everything I told you about was validated in Russia by Ministry of Health and Ministry of Internal Affairs. So we have certificates and everything.
Certificates from Ministry of
the Health
of Russia.
Because they provide the registration certificate.
Forensic HID validation.
ISO 18385 certificate is there?
Yes?
Yesterday.
I saw we have 1835185. Yes, yes, we got it. You know, in Russia, we say better one time to see than 100 times to hear. So you can see our equipment in the work in your labs if you like. We just come and show. No problem. Thank you.
So thank you, Iger, and the whole Sintol team. And thank you for wonderful presentation, and thank you for clarifying all the questions. Thank you so much.
Thank you.
Thank you all. Now I request to all our guests, please go and visit the gallery for the posters, and after that, proceed for the lunch. Thank you so much.
Respected all dignitaries, I have one announcement with the permission of Dr. S.K. Jain sir. Today, after this session, we are going to have panel discussion, and for that, we want that all the dignitaries who has come from different states may form a group, like who have come from north, who have come from south, who have come from northeast, who have come from west, so that problems can be addressed accordingly. So it is a request, please, to form a group in between us. Whoever has come, please write down their names accordingly, like who has come from the west side, who has come from the northeast side, who has come from the north side, so that we can address the issues during the panel discussion. And 5 minutes, little bit delayed because of the guest chairperson is just he is stuck into traffic. He's just coming within 5 minutes. By that time, you please form a group. Thank you. Respected dignitaries, participants, I would like to start this session with the permission of Dr. S.K. Jain sir. As our chief guest is running a bit late, but although he will join, since then we will start the session so as to avoid the wastage of time. For that, I would like to invite Dr. Hirak Ranjan Das to please come on dais and take over the session. I would like to invite Dr. Vinay Kumar, our distinguished guest and guest speaker from AIIMS Patna. Today he will describe about the medico-legal practices in collection of evidences, mainly related to DNA and biological samples. Followed by the address.
We are also privileged to welcome our distinguished guest speaker, Dr. Vinay Kumar, professor at AIIMS Patna. Sir, we request you to please come on the dais. To welcome Dr. Vinay Kumar, sir, we would like to invite Ms. Seema Patel, Assistant Director, FSL Patna. Dr. Vinay Kumar is a pioneer in medico-legal sciences, having initiated narco-analysis and psychological autopsy program at AIIMS Patna. He served as medico-legal consultant to the Bihar Human Rights Commission and provided expert services to CBI, CID and Bihar Police. Sir has authored 46 research papers, a book, contributed chapters on human rights and forensic psychiatry, and received commendations from the Bihar Human Rights Commission. Thank you, sir.
Thank you very much to the organizers for inviting me in this August gathering of forensic scientists from not only from all over the India, but also across the country, across the world. Now, actually this is the mainly conference of forensic science, and I am from the background of forensic medicine. What basic difference between these two, which I see, is in forensic science various divisions are there. Toxicology division, DNA division, serology division, so on, various divisions are there, for which specialized and qualified persons are working in particular division. Whereas forensic medicine people are the people who is having the knowledge of the medical science. And in medical science, nowadays at least 40 subjects are there. And around from all 40 subjects, specialized subjects, super specialized subjects, whatever the portion of those subjects which is applicable to the administration of justice has been compiled, and one subject has been formed, that is known as the forensic medicines. So what I see the basic difference between the forensic scientist and forensic medicine experts are, the forensic science people of the particular division is the master of that particular division. Whereas contrary, forensic medicine people are the master of none, but jack of all divisions of the medical science, whether anatomy, physiology, biochemistry, pathology, pharmacology, medicine, surgery, gynae, pediatric surgery, so on. And only those and respective portion of those subjects which is applicable in the administration of justice. So our main role is to give the doctors of a specialist and super specialist doctor to confine to his clinical service and patient care, and their medical legal part, their pain of the medical legal part, going to the court, everywhere, we actually observe. So we are the interface between the administration of justice and doctors. And all SOCs, legal SOC, people want to go to the court and have the concern with the police. So those legal SOCs, we are the SOC absorbers for the doctors, and those legal SOCs. So definitely, so when invitation came to me from the organizer, then I told them that this is the conference of the expert, master of ones, and I am the speaker of Jack of all. So he told me that, okay, the Jack of all has different role and master of one has different role. Master of one has depth of knowledge of work in one subject, whereas the Jack of all has superficial knowledge of the all other subjects. So in the optometry, we can see the master of one has deeper penetration of visualization, but field of vision is tubular, whereas Jack of all has field of vision is wider, but superficial penetration. So with this background, I am going to start my presentation. On medical legal case procedure. Definitely most of the biological materials comes from the hospitals, also from the scene of crime also, hospitals and forensic medicine departments. So most important thing is hospitals is not only receiving the medical legal case. Hospital is first meant for the patient care. And many patients comes to the hospital in which a doctor has to identify or has to suspect that which case is medical legal case. And that case has to be branded as medical legal case. And most of the problem arises here. Sometimes initially every case looks like it is non-medical legal case, and when time passes, evidence is lost, then people came to know, no, now it is a medical legal case, and the evidences which is lost means lost. So the most important factor in this practice is to identify whether it is a medical legal case or not at various entry of the patient at various starting point of time. Although there is no legal definition of the medical legal case, various textbook, various agencies, various organizations coin its own definition. So we shall not go into the detail of different definitions. I have chosen one very simple definition of medical legal case, very short definition of medical legal case, that a case of injury or illness where investigation by law enforcement agencies is required to establish fact and fix the responsibilities. So even illness is medical legal case. If suppose the person died due to disease. So till the fact whether there is a question that whether it is actually died due to the disease, or it is a case of homicide, or it is a case of suicide, or it is a case of accident, then the role of doctor or forensic medicine expert is just to differentiate whether it is died due to disease or it is died due to the foul play. Here role is simple. But if further in the illness, if the claim is that, okay, the patient died due to the disease, but died due to the negligence of a doctor in treatment. The same thing, whether it is clear that it is not a case of suicide, whether it is not a case of homicide, not case of accident. But when question comes with due to negligence, the same case which is non-medical legal case, only on the basis of one FIR, only on the basis of one complaint, only on the basis of one statement, it is converted into a medical legal case. Then the role of medical legal experts shall not be limited to give the opinion that whether it is a case of death due to disease, but we have to investigate further which disease was, what was the exact condition, what was the situation, what was the management. Then entire investigation, entire spectrum of the opinion changed. In simple case, whether it is a natural death due to disease or in there is a foul play, we simply say, okay, the patient died due to the diseases of lung. This opinion is enough to stop the case if question regarding medical negligence is not there. But as soon as in same case, once question arises that it is a case of medical negligence, then we have to further inquire, then we have to further diagnose. If it is a disease, then what type of disease, what was the condition, what treatment given, everything comes in the picture. So that's why it is very tricky issue to decide whether it is a case of medical legal case or it is a non-medical legal case. However, by convention, as we all of you know, these are the examples of cases where at the entry into the hospital, people brand it is a case of medical legal case, and in this case, police has to be informed by the medical officer and further police. And in this last 2 point is very important. Other points are very clear. Last 2 point is very important, that any unknown or unconscious patient, if comes to the hospital, then it should be branded as a medical legal case. And besides this, broad dead or death occurring within 24 hours of the hospitalization without establishment of diagnosis. So if this criteria comes, the case should be branded as a medical legal case. Okay, further investigation shall be done. If it is not a medical legal case, it shall be branded. But other are clear, but these two things, last 2 point is important, and most of the mistakes happens in these in last 2 points. So it has to be taken care. And very important point is suspicion of legal relevance is enough to register the case as MLC. Means it is not required that some proof has to be given that to brand a case as MLC. Simply a doctor has any suspicion, so only suspicion is sufficient to brand any case as a medical legal case. Now, as discussed, what is the importance of branding a case as a medical legal case? If medical legal case, a case which is actually medical legal, but by mistake it shall not be branded as medical legal, then what problem shall happen? This medical legal case ensures justice delivery, because once medical legal case shall be branded, then all medical legal investigations shall be done. All medical legal points has to be taken. Protects the right of the victim and accused both definitely. If the accused is innocent, in our state, the actual case is less and false cases are more. That is the reason of low conviction rate. So it is beneficial for the right of victim. Many situation comes where near and dear comes to the hospital. So the doctor, there is no issue. That's just heart attack, problem of heart attack is there. So sometimes in good faith, doctor and patient comes unconscious, doctor in good faith, they do not brand it as medical legal case. Later on some other relative appears that no, it is a case of medical legal case. So in that situation problems happens. Preserves the scientific evidence. If we shall not brand the medical legal case, scientific evidence, we shall not preserve the scientific evidences. Supports the court testimony definitely. Reduces the chances of evidence contamination and rejection. Problem what happens when patient comes, the primary aim of the doctor to save the life of the patient. And during the life-saving procedure, many scientific investigations are lost, scientific evidences are the lost. And definitely, so if already bit red color medical legal case is branded, then everybody shall be cautious about the preserving the scientific evidences and enhance the credibility of the medical practitioner definitely. Now, principle of the medical legal procedure. Once medical legal case is branded, then what happens? There should be promptness in the examination and documentation. All attempt has to be done. Okay, treatment is the most important part. Even at the cost of losing the evidence, treatment cannot be stopped or but definitely another team should continue to record the medical legal evidences. Neutrality and objectivity and informed consent. In medical science, the chapter of informed consent, entire book has been written on the informed consent because we are dealing with the human body and a human highest animal of the earth. So lot of rights and lot of everything is there, lot of law is there. So informed consent means it is said that the consent has no meaning if regarding the procedure, regarding the condition, the explanation not given to the patient. So informed consent, non-informed consent means no consent. So it is the importance of informed consent. But some exception I have, according to the law, here we have highlighted. Under section 51 BNSS, arrested person may be examined by the registered medical practitioner without consent by applying reasonable force if requested by the police officer not below the rank of sub-inspector of the police. So some liberty has been given here in medical legal examination of the accused patient who is not giving consent, but sub-inspector of the police or police officer at the level of sub-inspector or above the level of sub-inspector of police thinks that no, examination is necessary and we should utilize the reasonable force also for the examination, then it allows the examination for that. But here topic, especially with the DNA, samples for the DNA collection, whether this 51 BNS is applicable or not. According to the BNSS, verbatim of it is applicable to this procedure also, collection of the sample for the DNA also. But different courts has passed different judgment. No, it is the violation of Article 23 of Constitution of India, etc. and etc. So practically there is a restriction in applying the section 51 BNSS in the collection for the in the collection of the samples for the DNA analysis. Therefore, what we practically, and there is no clear, the court judgments and BNSS is contradicting each other. So in this way, the safer side is, particularly in the situation of DNA analysis, if the person gives consent, it is okay, person can give consent. But if person is not giving consent, usually investigating officer should take permission of the court, take direction of the court that in this event force should be applied and collection of DNA material is necessary, then court permission, by court permission we can take. It is the practical approach. in the situation where contradiction is there in the bare acts and the court orders. And it is further stated that if the accused or victim both is female, she should be examined by the female registered medical practitioner or under the supervision of the female registered medical practitioner. Sometimes, suppose a specialist is not female, but we keep the non-specialist female, a medical officer who is female, in the presence of that female, a male can examine. But female registered medical practitioner should be present according to this BNSS. But there is no law to examine the victim without consent. No court, no law can compel any victim who is especially here we mean the victim of the sexual assault, especially mean. No court, no law can compel the victim to undergo medical examination or medical legal examination without consent. Medical examination without consent may be done in emergency situation. For that, another section of law is there. To save the life of the patient, and if nobody is to give the consent, and person is not in the position to give the consent, then for the treatment purpose, medical examination and treatment can be done without consent. But as far as medical legal examination is concerned, for the investigation purpose, no law can compel the victim to undergo the medical legal examination without consent. Then confidentiality. There is a law, universal law, that every communication made between a doctor and patient or examinee and examiner is confidential, unless law provides especially, and that exception we here use the term privileged communication. So on the privileged communication, very big chapter is there in medical law and ethics, but here I want to say only the word. In the situation of privileged communication, we can divulge the information of whatever we received during the consultation to the appropriate authority only. And all of we know that during disclosing the identity of the victim of sexual offences is punishable under section 72 of the BNS, which was former 228A of the Indian Penal Code. So that is same in the POCSO also. Accurate documentation, definitely. Proper evidence collection and preservation, definitely. Maintenance of the chain of custody, very important. In our state, day-to-day problem of maintaining chain of custody is there, due to which the defense lawyer take advantage, due to which many tampering of evidences may occur. So chain of custody. So the stepwise MLC procedure is registration of the cases of MLC, most important. First point, it is most important point to take in immediate decision whether this is a case which is a MLC case or it is a non-MLC case. Then inform the police where required. And regarding information of the police, various law is there. One section 31 BNSS includes the list of offences which the public to give information to the nearest magistrate or the police officer. Earlier it was section 39 CRPC, and it is section 31 BNSS. This 31 BNSS contains the list of offences in which, if anybody came to know regarding those offences, whether it has been committed or about to be committed, every public, including medical officer, has to give the information to the police. But however, cases related to sexual offences 63 to 79 BNS is not included in the list. Now, lot of hue and cry, police raises question that they think that, any type of offense which the medical officer or any public came to know, they have to inform the police. It is not true. Only those cases which is listed in section 39 CRPC or 31 BNSS, offences regarding related to those offences only, it is necessary to inform the police. However, there is various paraphernalia is there ki how this any case can be made under that law. That is the art of the police well know. So people take safer side. But rule is that. So sexual offences cases, but a case under POCSO has to be reported to the police. But if that woman of victim of sexual offence who is below 18 years of age, then it is necessary to inform the police. So where the practical utility of these two laws is there? That is why if a major above the age of 18 years, victim of rape come for MTP, medical termination of pregnancy, usually they contact doctor for the medical termination of pregnancy and due to rape or other medical examination and do not wish to inform the police, police should not be informed. So utilizing this law, we have to maintain the confidentiality. If a victim of sexual assault comes to a doctor, comes to a specialist, says, this pregnancy is due to rape. The specialist says it is due to rape because there are criteria under the MTP Act, under which condition MTP shall be done and under which condition MTP shall not be done. So in rape, the MTP Act allows termination of pregnancy. So if woman comes or a girl comes that this pregnancy is due to rape, then every registered medical practitioner had to terminate the pregnancy if she wishes. And if she wishes that I said, oh, it is due to rape, but I do not want to file FIR, I do not want to inform the police, all these information has to be kept secret, then a medical officer or person who is conducting the MTP should keep the information secret. There is no need of informing the police, but definitely there should be a written statement. A doctor should take written statement, all those that this pregnancy is due to rape. I do not want to inform the police. I only want to do the MTP, medical termination of pregnancy. Police should not be. But at the same situation, if she is below 18 years of age, but whatever her wish is there, a doctor should not honor her wish. If below 18 years of age, a doctor under POCSO, doctor has to inform the police even against the wish of the that girl and woman. Regarding that, even it is not required. Even if say one liberty has been given here also in the law, which I have not mentioned in the slide. If there is a borderline, if there is a doubt where that girl is below 18 years of age and or that girl is above 18 years of age, and if that girl is saying that no, I am above 18 years of age, I want to, or even doctor को doubt भी लग रहा है, if doctor no, she may be 17 years old, but he has to accept the statement of that girl and he have to see for the purpose of MTP, doctor has to accept the statement of the girl and should proceed for the medical termination of pregnancy. So under MTP Act, so much liberty has been given to the woman. And okay, but if there is a huge discrepancy, it is found there is some, then definitely examination may be done and police may be informed, because doctors should also be very sure that whether there is no borderline cases. So it is especially for the borderline cases. And failure to inform the police whether it is required, punishable under section 211, 238 and 239 of BNS. So BNS, sorry, I have by mistake I have mentioned here BNSS. It is only BNS. Last point. Now, obtained identification and relevant history after following all these rules. Consent for examination and evidence collection, as discussed that for medical examination purpose, separate consent has to be taken, and for medical legal examination, separate consent has to be taken. Patient may come for the treatment, but they don't want medical legal examination. If she refuses medical legal examination, she can refuse, we can't do medical legal examination. But definitely written refusal should be obtained in the case sheet, and then only we can proceed for the treatment and medical legal exams. Then clinical examination and treatment, definitely it is the most important work at hospital. Even if the person is under threat of life, leave the medical legal examination. First importance is to treat the patient. And first the stabilization of the patient should be given priority over all works like medical legal examination or police information. However, in ideally multi-specialty institution, bigger institution like AIMS, in AIMS Patna also we follow that a separate team of medical legal experts are there usually in the emergency to record the medical legal experts are recording the medical legal evidences, and clinical experts are involved in the treatment of the cases. So side by side, both work are done. But treatment is priority. Even in one case, I went for the giving evidence in the court. So defense lawyer said that at the primary health center, doctor only noted only 4 injuries, whereas in post-mortem examination, they referred the higher hospital whether died, and you in the post-mortem examination, you are recording the 15 injuries. So there is contradiction. Defense lawyer want to take contradiction. Then I replied that their primary aim is not to record medical legal examination, not to record medical, his or her primary aim was to save the life of the patient. And because appropriate facility was not there at the primary health center, so his primary importance was that after stabilizing the patient, as soon as possible, they have to refer the patient to the higher center. So there is quite natural that there is difference between the injury report of the primary health center and injury report of the higher center and post-mortem examination report. This difference is quite natural. So that has to be explained in the court of law. So definitely detailed documentation and finding, collection of medical legal samples, preservation and sealing and labeling, proper labeling. ना आप लोग बार बार पुलिस गलती करके आती है, लेबलिंग के चलते वापस भेजते हैं. So proper labeling definitely, otherwise tampering of evidence may occur. It has to be, it is definitely required. Handing over to the investigating officer and authorized agency, and preparation of the medical legal reports. So this and detailed documentation of the injuries, we are, when still I am medical legal consent of the Bihar Human Rights Commission, and I have to review all the cases of custodial death occurring in a state, and when we see the medical records, when we see the post-mortem reports, etc. So everything, shape, size, color of the injury, type of injury, what is possible of the weapon of the injury, every detail has to be written, but due to absence of these information, later on we are handicapped to give any opinion whether it has happened. In many cases, simple injury due to unintentional injury becomes serious issue, and we have to give opinion adversely. So maintenance of chain of custody, definitely we have observed many cases where only due to absence of chain of custody cases, I shall not give the example, and because many things are prejudice, but it is the situation. Registration and documentation, what are the information required in the registration and documentation in record? So patient name, age, sex, address, date and time of arrival, brought by whom, police information if available, and alleged history in the patient's own words as far as possible. So patient's own words is important. If the person is not able to give the history, then other person may give history who know, and there is another situation of concept of dying declaration, everybody knows. So history. And these all things has to be entered in MLC register in the hospital. MLC register is the separate registered, and assign MLC number. So different medical legal case number is assigned, and important is it's avoid interpretation at this stage. During the recording, only recording we shall make. Later on, interpretation shall be done, opinion shall be done, and never alter record without proper correction procedure. So many times we see the cut overwriting is there without initial, without signature. So it has to be avoided. Consent in MLC and DNA sampling, medical examination required, as discussed, medical examination required informed consent. As discussed, informed consent is very big chapter. As well, even for one procedure, all of we have witnessed before surgery, सबके घर में कभी न कभी सर्जरी हुआ होगा तो सर्जन टेक्स कंसेंट की एक लाइन में ऑपरेशन एवं बेहोशी के हिंदी में ऑपरेशन एवं बेहोशी के खतरे को जानते हुए मैं अपना मरीज सुपुर्द करता हूँ. दिस इज नॉट द एक्चुअली इन्फॉर्म कंसेंट. नाउ हायर कॉर्पोरेट हॉस्पिटल्स मेक्स द बिग information, but that is also too much information. Corporate, actually they want to do the all entire fact chapter of the textbook. Suppose you shall go to the hospital for MI, suppose angioplasty. So regarding angioplasty, whatever chapter written in the book, entire chapter is printed on the consent form, and there they take signature. So much defensive era of the defensive medicine is there. That is also not required. Required is that you have to give information those which is common problem, common side effects, and some side effects which may not be common but serious side effects. So combination of both is necessary. These information should be given necessary. And definitely minor or mentally incapacitated person, consent from the parent, guardian and legal authorities is involved. Regarding the age of the consent, I have not mentioned here, but 3 borderline is there. If the child is below 12 years of age, he cannot give consent for any procedure, any medical procedure or surgical procedure. If it is between 12 to 18 years of age, the child or that person can give consent for only those procedure which is for his benefit. Those procedure which is for his benefit. And above 18 years of age can give consent for any procedure. For example, very simple example is there, that if any person has admitted in the hospital and no one is there, and that person is in between 12 to 18 years of age. So the treatment is being done for his benefit. So a person between 12 to 18 years of age can give consent for treatment, management. But if suppose a person has to give organ donation, it is also medical procedure. So blood donation, blood transfusion, blood donation, although it is medical procedure, but he is giving the blood for the benefit of other, not the benefit of self. So this procedure in between 12 to 18 years of age, he cannot give consent for this procedure, for the organ donation or blood donation. He can give, if a person is above 18 years of age, then only he can give consent for the organ donation or blood donation. So this is the law related to age and related to the consent of the procedure. And a refusal must be documented. If anybody is not willing, we are in the hospital facing daily issues. Even for his benefit, they are not ready to undergo the procedure. And if for every procedure consent is required, for that purpose we take the informed refusal. We record that, okay, the patient is not ready for this procedure, and we take signature of the patient ideally, and then otherwise many situation occurs later on. हमारे यहाँ legal awareness is so much there that lawyers are roaming around the hospitals. Any untoward happen, they are very much to pursue the case, no, file the case, so that whatever the compensation shall be given, 50-50. I shall not take no fees, 50-50 compensation may be granted. So for that procedure, doctor has to document everything in detail and every step, so that to prevent the allegation. Key point is that treatment should not be delayed due to medical legal formalities, and suspicion of legal relevance is enough to register the MLC case. So suspicion is enough to register the MLC case. No proof is required. Now, DNA evidence. Regarding the DNA, I have written a legal quote in my mobile. I have not mentioned in the slide. That it is said that regarding the importance of DNA, it is said that DNA profiling is the most objective, unbiased, and practically accurate technique in comparison to other technique of examination in the criminal investigation. So it says that in entire criminal investigation, various scientific and non-scientific test and examination is being done. And people's compared different examination in which it has branded DNA profiling is the most objective, most unbiased, and practically accurate technique in the crime investigation compared to the other evidences. So that's why this conference of the DNA and biological division is important, because you are dealing with the most legally most unbiased and most objective evidences. So DNA analysis, definitely these are the basic things. Helps in identification of the individual, linking suspect, victim, and crime scene, whatever the biological material, we can link the suspect. These are the basic things for you people, but I have to repeat. And sexual assault investigation, it is important DNA. All of you know, there is no need to elaborate. Paternity and maternity dispute, missing person and disaster victim identification. Nowadays, era of disaster victim identification is important. Identification of decomposed and skeletal remain when fingerprints are lost and all other situations are lost. So DNA is here important. Advantage is highly sensitive and highly individualized, highly sensitive and highly specific also, this DNA evidence. Very less investigations are there which is highly sensitive also and highly specific also. And in day-to-day Bhaiva, in the examination, MBBS examination, MD examination, I ask common question to the students, which is more specific, DNA, fingerprinting, or dactylography? Fingerprinting. The answer is always, it is the dactylography. Because in monozygotic twins, the dactylography shall be different, fingerprinting shall be different, whereas DNA shall be the same. So in this, the examination costing 10 to 15 rupees is more specific than DNA examination, which is costly investigation. But all of you are aware with the newspaper reports and social media report regarding this. Useful even for the minute biological material. This is even very small material. It is useful. Now all of you must have heard about this report. In Kanpur, from Kanpur it is reported that even one monozygotic, two brother who was the monozygotic twin, their fingerprinting also recorded as same. Even retina scan was the same. So this is the rare example, and theoretically it has been given in our textbook also that one chance in 64 billion population. There is one chances in 64 billion population where fingerprint of the person, two person is same, which is the above the world population, but within the world population, and in India also, and in actually regarding fingerprinting dactylography, every milestone is achieved by the India, starting from the invention and starting from the ending into the rare example of this twin baby. So, these things are, I think, very basic thing for this August gathering, especially for the DNA people. The common source is blood, buccal swab, saliva, semen, vaginal and cervical, anal, hair roots, nail clipping, tissues, bone, teeth, skin cells from the test object, clothing of the stand. All of you are the expert of these things more than us. Types of DNA sample from living person and types of DNA sample.
Dr.
from
Vinay, thoda sa, if you could
yes,
sum
yes.
up as earlier because we are past.
No, I was about to say that my lecture never stops. Somebody has to interrupt. So, thank you very much for interrupting me.
Considering your
and these things
resourcefulness in the topic, I think we have to little bit stick to the time.
So, definitely, I think medical legal part I have covered very elaborately, and DNA part, I think these are the basic thing for this gathering. So thank you very much. I think nothing new is there here. So take home message, that means conclusion. MLC procedure requires scientific accuracy and legal awareness. Early and proper evidence collection improves the forensic value. So early branding and early evidence collection. DNA sample must be collected in a strict contamination control. Where we are doing better, we can boast. From everywhere, from simple water, DNA material comes, but biological material, especially fetus, which we preserve in AIIMS Patna, we especially prepare distilled water. Thereafter, by a sterile common salt, we prepare the sterile solution, and that biological material we put there. Besides the dried material comes here. Proper leveling, sealing, and chain of custody is essential. Medical care of the patient remains the first priority. Repeatedly, it is said that medical care is the first priority. Secondary priority is the medical legal examination. And good documentation is the backbone of the medical legal practice. So the success of medical legal investigation depends greatly on the medical professional's ability to examine, document, collect, preserve, and transfer evidences in a scientifically valid and legally defensible manner. And proper handling of DNA evidence with the MLC framework is vital for the justice. I think these are my references, and these are the many thanks. Thank you very much.
Thank you, Dr. Vinay, for excellent presentation. You have covered almost all, basically the general principles of medical legal procedures. You have been very brief on the concept of informed consent, what it is role, what is the relevance of that. You have also mentioned about various sections of BNS and BNSS where the role of informed consent has been defined. You also had a brief passing remarks on the interpretation of what cases to be reported as MLC cases and what not to be reported, especially relevant to POXO case and in case if the survivor is more than 18 years of age. And rightly to conclude, you have also mentioned about the importance of DNA and the recent case which has happened in Kanpur. So the role of DNA and everything, biological evidence, we all know. My question little bit, if you could elaborate on the role of identification of DNA as much as snake bite envenomation is concerned.
A snake bite?
Envenomation, because routinely हम लोग विसरा snake bite में preserve कर देते हैं, we send it for FSL. Then sometimes we get report, sometimes we not get report. But what is the relevance in case if they are able to identify the animal DNA related to a case of snake bite envenomation?
Definitely by the common concept, I think, like human bite. And I have also forced the cells, I think, on the body when human bite, solving also DNA analysis is done. I think regarding the snake bite also, it should be same, but one problem is there, concept is there, ki a snake, in a snake bite, venom is there. Now venom is inside, till inside the thing, it is not toxic to the cells, but whether it has to be, I have not gone through in this in this thing, what venom, which is a poison, how it causes lysis of the own cells, which is poured on the snake bite. But even I think that during inserting the venom, still there are other saliva materials present around the snake bite site, and from that I think cells can be recovered. Animal cells can be. I think your question is whether this DNA belongs to a snake and or not, or some other person. And specifically, if this DNA belongs to particular snake, then which snake? But I think what is the capacity of our laboratory is that DNA is separated by target. No, it is not that. I think DNA sequencing is not done for the purpose of identity. Doctor Sahab, DNA sequencing is done for the purpose of identity.
Yeah, could you hand us mic, please? Because I think one case has been reported where they have identified the snake bite case.
So what I'm saying is it can be done, no doubt it can be done. They have developed markers for various snake species and it can be identified. But what happens, in case of snake bite death, individualization of the human being never comes disputed. Okay? It is not disputed. What do you want? Whether it is because of snake bite or not. If it is because of snake bite, फिर insurance मिलेगा उसको and he will claim his insurance, all these things. So for that, normally serological methods are widely available. I think most of these CFSs, FSS, they have the serological methods. They can identify cobra, either it is cobra or crate species or whatever. It is available. LH methods simply they are doing. Number two, the process of sampling in snake bite. What you do from MLC side, sir, you cut that person, put it in a saliva, salt solution, and then send it to the laboratory. So when you are cutting that particular skin, putting in a saline bottle, recovery of cell will be very difficult. But serological methods are, I think, widely available. We do it, sir. Consider no problem.
Yeah, thank you, sir.
I
Any
think
question from the audience?
Both swabbing is done, dry swabbing is also done, and normal saline, soaked in normal saline. Both are done, I think. Yes. Actually, in post-mortem examination, it can be done when a snake bite occurred, and without going to the hospital, already diagnosed dead at the site, and then comes to the post-mortem examination. Then it can be possible, but I think it is the requirement and capacity of the forensic laboratory, whether it is anything established in the forensic science laboratory on the basis of their use. कि कितना quantity of sample हमको मिलेगा.
One question from the audience.
Not question, I just want to add something.
Sure.
In CFS in Chandigarh, Dr. Rajiv, he has established a laboratory where using ELISA technique, he is able to identify whether it is snake venom or not. So he has examined 2 cases also. One is from Andaman. I am basically from Andaman. I am working over there. So we have sent a case of snake bite. So using ELISA technique, he told that this is from the snake. Exactly species he was not able to tell, but whether it is through a snake that was caused due to snake bite or not, this thing he has clarified.
That is also, sir, immunological examination.
Yeah.
Antigen antibody best examination. No?
Yeah, we'll take one last question.
Sir, it's not a question, sir. Just following to that, we have the facility here of ELISA, and we are performing tests. But, sir, actually what you has raised, the thing is that in many cases from district side, we receive viscera in case of snake bite. But in viscera, we could not get the venom, or even after running it on ELISA technique. We need that particular tissue only where the snake bite has.
And actually, sir, viscera is maybe sent for that. Actually, there is a question.
To exclude.
Question whether it is a snake bite, whether it is poisoning, whether it is other fall, other situation is there. For
Yes.
that, during examination, if it is diagnosed,
Yes.
then it is a note, it is a snake bite, not other than that.
Yes.
Then there is no, but most of the situation in case of
Suspicious death, right, sir?
suspicious
It may
death,
also
we
depend.
have to analyze after investigation actually what is there.
That the death has not caused
I think
due to
we
other
will
poisons.
Stop
sir.
on this. Acha, we will have one more.
As far as DNA is concerned,
Yeah.
we need cells.
Yeah.
Okay.
Yes.
If cells are there on the skin
can extract.
and a swab is properly taken,
Yes.
then we can isolate DNA. We can sequence also.
Yeah,
So this sample collection is very important in this case.
Yeah.
Actually, earlier also we were used to give opinion on the basis of clinical examination only. We see the two fang mark is there, and we straightforward give opinion it is death due to a snake bite. Now the advancement occurred, the process of ELISA test and other tests established in the FSL. Then we started giving the sample. So as the facilities are advancing, we shall start utilizing that facility.
I think we will stop here. A wonderful presentation and followed by a wonderful discussion. Thank you so much, Dr. Binay Kumar, and to the
आप
audience.
फोरेंसिक मेडिसिन के आदमी होकर के आप ही बोलिएगा वंडरफुल प्रेजेंटेशन, इनको बोलने दीजिए.
नहीं, ये तो एज अ चेयरपर्सन आपका प्रेजेंटेशन जो था उसको तो सराहना ही है, क्योंकि द इनपुट जो इनके हिसाब से थे बहुत इम्पोर्टेंट थे.
Okay, thank you very much. Thank you.
Once again, good afternoon, everyone. I'm honored to have with us our chairperson for today's session, Dr. Amit Patel, sir, HOD FMT AIMS Patna. To welcome our guest person, we would like to invite Shri Jitendra Kumar, Assistant Director, FSL Patna. Sir, please. Dr. M. Patel is the professor and head of forensic medicine and toxicology and associate dean at AIMS Patna. He holds an MD, DNB and LLB with specialized training in hospital administration and medical education. His extensive expertise includes conducting medico-legal autopsies, managing student affairs, and developing protocol for COVID-19 disease management. Currently, he serves as Vice President for both the Indian Society of Toxicology and Medico-Legal Association of Maharashtra. Dr. Patel is a prolific researcher with over 45 publications and has received multiple awards for excellence in forensic research and administration. He also serves as a key resource person for training medical officers in clinical forensic and immunization related deaths. We are honored to have you here, sir.
We have our next speaker, Dr. Irek Ranjan Dash, and he will be speaking on attribution of non-convoluted peaks in human STI markers and identification of its possible source.
Good afternoon again. Good afternoon again after 24 hours. So, No, sir, 24 hours. Okay. The topic I have chosen here is attribution of non-convoluted peaks in human STR markers and its possible source, finding of its possible source. Here, all the DNA professionals who are working in a forensic DNA laboratory, they must be aware of this kind of a DNA profile, which is called as an ideal DNA profile. So what do we mean by an ideal DNA profile? When we are talking about ideal DNA profile, at any particular genetic marker, we are expected to get either a homozygous peak or a heterozygous peak. And when an ideal DNA profile is considered, all the peaks, they should be above the threshold lines, and all the heterozygotic peaks, they should have the equal peak height, and all the homozygous peaks, they should have the equal peak height. And another one more consideration is that the sum of the peak height of all the heterozygous peaks, it should be equal to that of the peak height of the homozygous allele. So that's called as an ideal DNA profile. But in our routine casework samples, we do not get an ideal profile. We know there are many routine artifacts. They may be originated due to the routine PCR processes or capillary electrophoresis processes. They may be routinely called as PCR artifacts or capillary electrophoresis artifacts. The known established artifacts, they may be your noise, dye blobs, spike, pull-ups, stutters, non-template additions. We get such kind of artifacts in our routine forensic DNA casework analysis. What we do during our analysis process, simply we delete those attributes or those artifacts, commonly known artifacts. But in the process of doing those modifications to a pristine DNA profile, we need to give it a cause why we are deleting it. We may attribute what is the reason why we are deleting an additional peak which has generated in a DNA profile. Why I am saying so, why we should attribute a peak which we are omitting or which we are deleting, because the profile data is auditable. Always this profile data is auditable. Whenever we are doing any kind of modification in the generated data from the gene mapper IDX or after capillary electrophoresis, the data is editable and it is auditable. Whatever the modifications we are carrying out, that is being recorded by the system. Hence, for the deletion or modification of any observed peaks, we should give the appropriate scientific reason why we are deleting it. So proper attribution of a peak is necessary, but many a times we fail to attribute. Why I am saying so, I will give two cases which I did during my FSL days. This is a very simple case of sexual assault where no male DNA profile was detected. You can see the first row, it contains the DNA profile from the vaginal swab of the victim, and the second row, it contains the blood sample of the same victim. That is the reference sample basically. And in the amylogenin marker, as you can see over here, it doesn't give rise to any additional Y peak. That means it is not a mixed source. So the vaginal slide of the victim, it is generating a single source profile which is consistent with the blood sample of the victim. But one peculiar observation that we find over here is that you can see D18S51 marker. So except this particular marker, for all other markers, the alleles were consistent between these two samples, the vaginal slide sample and the blood sample of the same victim. But we do not know why this extra peak appeared. We may call it as a mixed profile because yes, the sample source is a vaginal slide, but none of the other markers except the 18S51, it showed the presence of any additional peaks. We fail to attribute what might be the reason of appearance of this additional peak. And to my misfortune, After few days, I was alerted with another murder case, and in that particular case also, I examined the cloth sample and the postmortem blood. Here you can see a consistent DNA profile was generated, except at a particular genetic marker, that is D5S818 again. You can see over here that allele, here it is 13 homozygous, but here it is 13 and OL. And OL, that is a commonly known artifact of ladder, but we cannot delete this OL. A forensic expert can easily identify that it is not an artifact, it is a very true peak, because the peak height of that off ladder peak, it is just equal to that of the peak which is being generated for the 13 allele. So we found no reason to delete that OL, considering it to be an artifact. So we are clueless what to do, how to report this case. And we need to attribute why this additional peak is coming. When we studied the literature, there are many possible causes of more than expected number of alleles which can be generated at a particular locus. It may be stutter products, as I was discussing earlier. It may be incomplete 3' additions, signal exceeds the dynamic range of instrumentation, poor spectral calibration separation, too much of DNA, incomplete denaturation of double-stranded DNA, or presence of exogenous DNA. So these are the literature says that these are the possible causes of generating an additional peak. What we did over here is that using that same set of DNA which was extracted from those samples, we amplified 16S rRNA gene. And while amplifying this 16S rRNA gene, we could able to get a proper peak. What does it mean? What does it mean? That means let me clear you that 16S rRNA gene, it is the housekeeping gene for microorganisms, mostly the bacteria. And here in our routine forensic DNA analysis workflow, we are conducting DNA extraction. When I am talking about DNA extraction, it is all type of DNA. It is human DNA, it is plant DNA, it is microbial DNA, it is animal DNA. It is all type of DNA. So these forensic samples, which are a rich source of nutrients for the microorganisms. Microorganisms, they need food for their growth. And these forensic biological samples containing protein, lipids, carbohydrates, and nucleic acids, they are the jackpot of food sources for the microorganisms for their growth. We are not conducting a human DNA specific isolation protocol. So I did a background study on this. Why this peak is coming? Yes, 16S rRNA is there. DNA from the microorganism is getting amplified maybe, but why? Yeah, all of us know from 1st January 2017 onwards, we are analyzing at 20 STR markers. Here the markers which are given in yellow highlighted, they are the old 13 codis markers, and the red one were the expanded codis markers. So I did a small study, small bioinformatics study. For most of the genetic markers, the primer sequence is available. I took 4 primers, THO, TPOX, CSF, and VWA. So the primer sequences were taken. After taking of these primer sequences, what I did, I identified the putative primer binding site. What happens? During the routine PCR process, amplification of any STR markers, the primers, they go and bind to the DNA. So, if any DNA is present which is having this primer binding site, that is expected to be amplified. So, I identified the primer binding site or the putative primer binding site of the particular STR markers. And you will be surprised to know, after finding of these putative primer binding sites, I did a blast. Blast means that's a, you can say, a Google search kind of thing. It's a bioinformatics tool where if you put the DNA sequence, it will show you the similarity to which organisms the similar sequence is found. That is in layman language, that is called as BLAST. And you will be surprised to know that this primer binding site was found to be present at a 100% blast identity score in Helicobacter, in Staphylococcus, Bacillus, Fusarium, Pseudomonas, Staphylococcus, Microcystis, so many number of microorganisms. That means, what does it reflect? If a biological sample is contaminated with so and so microorganisms and their genome is co-extracted along with the human DNA, then there is a possibility of amplification. If we look into the SWAGDAM guidelines, every kit is validated as following the SWAGDAM guidelines before their use, before their forensic application in the routine forensic DNA laboratory. Before their validation, they do validation in many aspects in terms of their sensitivity, in terms of their robustness, PCR tolerance, PCR inhibitor tolerance capability. They also study the non-human aspects. That means whether there is any non-specific amplification that is being there for the particular kit or not. So as per the SWAGDAM guidelines, in the non-human studies, they have included for primates, gorilla. That means, but what does it mean? That the kit which has been developed, it should not amplify DNA from gorilla, from chimpanzee, from macaque. Similarly, for the non-primates, they have chosen, they have put the guidelines to test the kit against mouse, dog, pig, rabbit, cat, horse, hamster, rat, chicken, and cow. Similarly, for the microorganism studies, the SWAGDAM guidelines recommended the use of Candida, Staphylococcus, E. coli, Neisseria, Bacillus, and Lactobacillus. But in our study, which I showed you earlier, except these two highlighted in red, that means Staphylococcus and Bacillus, the putative primer binding site was found to be present in such a huge number of additional microorganisms. Helicobacter, Fusarium, Pseudomonas. So if there is any chance of contamination from this particular group of microorganisms, then the STR markers, they may generate an additional peak. So we published this work in Forensic Science International Genetics in the form of a letter to editor, attribution of non-convoluted peaks in human STR markers and its possible microbial connection. So on this aspect, in this paper also, we recommended some suggestions. There should be proper attribution of artifacts while we are reporting a case. Microbial contamination can also contribute to peak generation in DNA profile, and inclusion of more candidate organisms in the form of microorganisms for the validation of commercial available kits. That's all from my side. Thank you. Thank you so much. If there is any question, I'm happy to answer. Yeah. Yeah, somebody is asking.
Hello. Sir, in one of the cases which we were analyzing, there was one situation of triallelic.
Triallelic, okay.
Yes, sir. So how do we distinguish between if there is any artifact or is it a triallelic?
Okay. So triallelic pattern, mostly you look into D21S11 Down syndrome. Actually, sir was saying also, if you look into the blood sample collection form, there also you can find the genetic disorders that have been reported. First aspect, from there you can cross-examine. And second one is the triallelic pattern at the particular STR markers, they have already been reported. And you see, if you are generating a DNA profile from a control sample or the reference sample, then it is not at all a problem. But here in these particular two cases, whatever I showed, I got confused in the question sample actually, not in the reference sample, because the reference samples, they are properly collected, properly stored, properly transported. So they do not create a problem. Only it is in case of the question samples where we get so many confusions, so many additional peaks may be generated. Okay, so triallelic pattern, it is already established in which STR markers the triallelic patterns are, can be generated. And there are two types of triallelic pattern also, type 1 and type 2. Type 1, I think, 2 peak height of both the alleles. If you sum them, then that will be the peak height of the third allele. And type 2 types of triallelic pattern, that means all the 3 peaks, they will be having the equal peak height. Okay, so there are 2 types, and it has been well characterized in the markers. Okay. Yeah.
Sir, I'm Yashwant. Now I understood the reason behind the peak.
Yeah.
So how do we incorporate that finding into our result? We rerun the test or we incorporate that finding as it is in our result?
Yeah. So always actually I used to say, when we are reporting a case, we should think what will happen to this case after 10 years. Because there are experienced Sarma sir is there, Mahapadra sir is there, they must agree with me that the case which we did in before 10 years, we are appearing for the court testimony right now. The technology has changed, the knowledge has changed, and knowledge is on the tip of your finger for the cross-examiners, for the defense certainly. So whenever we are reporting a case, we should think what questions might come after 10 years in this particular case. So we should know. Actually, it is science. We are a man of science. Whatever the finding is coming, we just need to report that. It may be a positive finding, it may be a negative finding. We must report that. And when the finding has come, what might be the cause behind that finding? That we need to explain to the court. What is the finding? What are the technology used? And what might be the cause for that finding? So when we are reporting a case, what are the limitations of this technology? That also we need to identify. That needs to be supplemented in a report also. So here, yes, we have identified an additional peak, and this might be due to microbial contamination as suggested by the amplification of 16S rRNA gene. That needs to be attributed properly. Thank you.
Your opinion. It should not differ, right? No. You write it in your observation set. If you are writing in your opinion that there is a time, then case is gone. See, scientific investigation, see, you are doing scientific investigation. Court is heavily depending on your opinion only. If you are diluting your opinion at any point of time, it is going to create a benefit of doubt, leading to acquittal.
Is reproducible many times, then we can say is a contaminant or triallelic. Reproducible होना चाहिए. We must repeat this sample again and again. Yes. जब तक रिपीट नहीं करेंगे, हाउ कैन हम क्लैरिफाई करेंगे कैसे कि वो रिपीट कंटामिनेशन से आ रहा है? सेम रिजल्ट आना चाहिए ना सर. रिजल्ट आएगा. It may be contamination, sir. It may be contamination. नहीं, ओपिनियन में तो मैं देने की बात ही नहीं कर रही सर. मैं कंफर्म करने की बात कर रही हूँ कि कंफर्मेशन होने चाहिए कि वो पीक कंफर्म हो रही है या नहीं हो रही.
I think this is a discussion which is going on. I think sir has already been.
पीक कंफर्मेशन की बात कर रही हूँ.
Sir has.
हमें नहीं देना उसको.
Sir, I think we'll close the session.
Sure, sir. Sure, sir. Thank you. Thank you so much.
Thank you so much. Thank you so much.
Thank you, sir. These talks were particularly enlightening and will guide many of us in our future endeavors. Now, we would like to call upon Shri Satyakam, SSA FSL Patna, to present memento to our respected Dr. Amit Patel. Now, we would like to call upon Shri Mohit Goswami, SSA FSL Patna, to present memento to our respected Dr. Binay Kumar sir. Let's move forward to the panel discussion session of Setcon 2026. I feel highly privileged to welcome our respected panel member, Dr. S.K. Jain sir, Dr. B.K. Mohapatra sir, Dr. A.K. Sharma sir, Shri Himjay Kumar sir, Dr. Devendra Kumar sir, Dr. Hirak Ranjan Das sir, and Dr. Amit Patel sir, and Dr. Binay Kumar sir. Sir, I request you all to please come on the dais and carry forward the session.
So coming to, ladies and gentlemen, please, may I have attention, please. Coming to the almost concluding session, the panel discussion. The aim of this panel discussion as suggested by Jain sir, will be to bring solutions to the problems that are encountered by the stakeholders. And who are our stakeholders? Are the FSLs and CFSLs. Now the problems will mainly be related to pendency of cases, because it is a DNA conference and we'll be talking about pendency in DNA cases in various labs. Next, modernization of lab, what effort you are going to take and what is your planning for modernization and upgradation of your laboratory. Amendment of recruitment rules so that new talents, new students, emerging students, they can be inducted in your laboratories. Improvement of workers, improvement in this sense, additional recruitment of additional workers, and funding. Of course, how to tackle with the new criminal laws. So what Sir has suggested, we can have the problems from all the state labs. We can divide it like this: northern states, eastern state, western state, and southern states. Please come forward and tell your problems regarding this thing. And the panelists, we have eminent panelists over here. We have doctors, we have experienced scientists, lab directors. So they will find out some solutions, right? So anybody from, otherwise I'll call you lab. Anybody from Rajasthan side? We can call lab by lab, sir, if they have. I have the pendency list with me, sir. And mic.
Good afternoon. We are audible? Yes. Yeah. So, this panel discussion consists of the experts from various fields, from forensic science, from academician, from medical legal experts. Now, the basic point of this discussion is, in our forensic labs, the case pendency is very high. Number one, in DNA, as per the new criminal law, especially in the POXO cases, we have to give the report within a period of 1 month. But in some labs, the cases are pending more than a year. So, what are your plans for reducing this pendency? What you need from the DFS headquarters, and where are your gaps? This is the major concern for discussion. So we have divided into 4 major groups. One is northeast group, another is north group, western group, and eastern group of the labs. So I have the status of the pending with me. If I see in the Rajasthan, who is from Rajasthan? Somebody from Rajasthan? Some officer from Rajasthan here? Can? नहीं है. राजस्थान से कोई नहीं है. MP से? Somebody is from MP? Yeah. So in MP, the pendency of the cases is 19,500 at present, as per the data provided by you. In the DNA cases, but
please stand up and talk, ma'am.
This is for all state, all labs, and from UP. Who is from UP? In UP, there is a pendency of around 15,000 cases. So Yeah, in all 7 labs. Because this data is provided by your lab. So our plan is, have you identified the idea or your plan of work, how you will reduce this dependency?
Sir, the major problem is in our labs is labs of human resources. Main problem is this. Secondary is the consistently supply of kits and chemicals. There are so many labs in between, before 2 or 3 months, there was problem. There was lack of kits in particular labs. That's why the scientists have stopped their case working. That was a problem. After a few months, when we get a consistently supply, they get started. That's why I mean to say there are 2 main problems. We have both problems. We have lack of manpower, human power, and along with this, we don't have kits and chemical consist supply. Secondly, sir, one more thing, if you allow me to say, one oversampling is the major problem in my state, especially from medical side. The accused in such cases where the gap of the medical and time of crime is more than even 2 days or 3 days, in those cases, the samples collected from the accused are even 10 to 20 from each and every. If there is 2 or 3 accused, we get 60 samples, unnecessary, from the accused. What we have to do? These are the problems, sir, mainly.
I think such problems will also with other state also. I understand. So there are some problem. So as from the UP state,
yes,
you have any plan how to overcome all these problems?
Sir, recently we are planning to have P model to get over our pendencies. That's why in this, our DGP sir has given his consent. And we are going to start, maybe started, as a pilot project in our Lucknow lab. As there is more than 6,000 pendency in our lab, it is expected that we are trying to have 2 years target to finish it up to 0%.
Okay. So what about your filling of the vacancies? Because ultimately the report will be issued by a government official.
Sir, we have up to 75% vacancies.
Yeah, that's why I'm telling.
Sir.
What is your plan?
Sir, we are just...
Until and unless you will fill the vacancy,
sir,
this PPP model will not work.
Sir, yes, sir.
Even in any lab. To report to be signed by the government official.
Actually, sir, we are we are categorizing our manpower in units. And nowadays, we have 21 units for DNA labs. And if we talk about the pendency P model, we are creating a unit having 1 RO reporting officer, 2 scientific assistants, and 2 helping hands. This unit will do up to 50 to 60 cases per month with the help of P, in which the case registration, receiving, and sampling will be done by our FSL staff. And then after the further steps of extraction, quantification, amplification, and fragment analysis, that all will be done by, it is proposed that it will be done by the private firm. And when they will give us raw data, our reporting RO will go for its reporting purpose.
But because so many biology experts are also here,
sir,
and I think we should revise it. This model needs to be revised. Reason is,
sir,
and DFS in collaboration with NFS is conducting the fact and fact plus exam every year, forensic aptitude and calibration test,
sir,
in different disciplines, in biological science, chemical science, physical science, cyber forensics, and psychology.
Sir.
So every year we are conducting. You can hire those people on contract, and they are trained also.
Yes, sir.
And against your vacant post, or your state can create some funds for those professionals, because posts are already sanctioned in your state.
Yes, sir.
And these contact people can be taken in a speedy manner also. The same model can be adapted by other state also for filling of the vacancies. If you are disagreeing my suggestion, if somebody add to something, I am ready to listen from those side. But this is our suggestion from DFS angle. Till your vacancies are filled, you can take forensic professionals from Fact and Fact Plus for this area. As regarding that model, I really appreciate, as you suggested, this model, PPP model, you can take some third party, they will give the equipment, they will supply the chemicals,
Chemicals, yes.
and there may be contract for sample basis.
Yes, sir.
Okay. Similar model can be adopted by other labs where pendency is very high. So that we can bring the pendency. And I have another suggestion also, and you can also inform your director.
Sir.
Because some senior scientists are retired or going to retire within 1 or 2 years. You can also engage them on contract basis, pay minus pension.
Yes, sir.
So this can also help you in reducing the pendency.
Yes, sir. The correspondence regarding...
This is the government view. First and most important aspect is our report should go within a month. This is the basic concept of this conference. Our pendency should be 0. Some are big state, their case inflow is very high. Some are small state, case inflow is very less. If their case inflow is very less, they can manage. But state like UP, Rajasthan, MP, Karnataka, Maharashtra, and Odisha, like this, where the pendency is more than thousands, so we are considering these are the biggest states. And another thing we are also proposing, all labs should come for accreditation.
Yes.
If our report quality is not good, we cannot stand in the court of law. As in the morning there was a session, quality reporting and uniformity, and Dr. MJ was presenting that. But 3M are important. I am always telling 3M, man, machine and method. You should have qualified man power. You should have procedure, method, should be well validated, and machine. Machine should also validate. I think all the lab have the validated methods, validated machines. We have already developed the SOPs, and purchasing of the equipments also be where the list of the equipments, standardized list, and as a MHA, DFS, we are also giving the funding to the state to bring the uniform. So, and now the question is qualification and expertise of it is not uniform as I understand. For that, we are proposing there should be regular training and retraining of the experts. And forensic science laboratory CFSL we have designated as a center of excellence in biological sciences, and experts from different laboratories can undergo training from the June month we are going to start. And we will do the proficiency testing of the scientists also. The PT program is very important. And we will send the material in advance, they can study it. After there will be online sessions, and then they have to undergo for test also. This is our plan. So quality is very important, and I think same is required for all labs. So this is the one aspect, and we are proposing to you. If you have any suggestion or any modification in this aspect, please tell me, so we can accordingly modify it. It is not, it will be one-way traffic, it will be two-way traffic, because you are the user. We are only proposing you. So this is the thing. And regarding RRs, you have to prepare your recruitment rule also uniform, so that other qualifications can also, experts from other qualifications could also be registered. Because RRs are very old.
Yes, sir.
So if now the technology changing very fast. If somebody is BTech from biotechnology, you need those people also. Similarly, in engineering field areas can also be taken. So you can think over it. This is my suggestion. And if you have any point, we can discuss our panelists, or one by one you can ask from any question from us?
So regarding a first query, where she said they are getting a lot of samples in each case. Regarding first problem, she said they are getting a lot of samples. Now medical doctors are there. Sir, actually it's a fact. What happens when you receive, am I audible clearly, sir? When you receive, say for example, a sexual evidence kit, right? Normally 20 to 30 swabs will be there. High vaginal swab, pre-vaginal swab, low vaginal swab, again high vulval swab, low vulval swab, posterior swab, anterior swab. Is there any mechanism to reduce these swabs? Or is it essential to collect all 20, 30 swabs? Of course it is written in your book, I know, but because as a scientist, what they have to, उनका मजबूरी क्या है कि they have to analyze each and every swab.
Thank you, sir.
Am I right, ma'am?
Sir, before answering your question, I would like to answer the questions related to which sir has already discussed regarding the manpower in your inside the institute. And I can freely tell here because I think all scientists are there, technical persons all are there. When you say about the PPP model, the bureaucrats and politicians becomes very happy. As soon as they hear word PPP model. And in my opinion, the technocrats should be very sad and a scientist should be very sad on this PPP model. Yes, this model should be there only to manage the emergency crisis. Short gap arrangement only. In my opinion, a straightforward PPP model is the just tool for exploitation of the talent. So, yes, but situation is there, recruitment rule, recruitment process for the permanent recruitment is so lengthy and we have to reduce the workload. Therefore, in my opinion, okay, it may be the workable situation, but only for the short period, only to handle the crisis of the short form the emergency. Yes, one good suggestion is that engagement of the retired personnels. Nowadays, medical science has evolved so much that life expectancy is increasing. So even after retirement, many persons become very young, healthy, mentally active. And the talent of the experience of those people has to be utilized. So I think I am in the opinion that this mixture of PPP model and this senior retired people, because when retired people joins the system, then you give the money pension and pay minus pension is the fact. But if that person shall be recruited on contract and purely on outsourcing basis, so they shall get advantage, they shall get full pension because he is coming on the PPP model. So and they shall take the full salary. So I am in the opinion that in the favor of the scientists, scientific fraternity, that this mixture of the model should be better than the pure PPP model or pure traditional engagement of the retired person. So this is my opinion. Another thing regarding your question, the problem is that as soon as more number of YouTube and media channel shall be increasing, your sample shall increase. Because this is called in the medical practice, this is called the defensive medicine. Defensive medicine means that earlier doctors used to treat the patient on the basis of clinical judgment, on the basis of his experience. But as soon as litigation against the doctors has started increasing court of law, then there only one question defense lawyer asks whether this test was done or not, why this test was not done, how you decided on the basis. Because on the clinical judgment there is possibility of errors. So more and more litigations are coming, the doctor writing more and more investigations. Likewise also in forensic practice, earlier actually on the medical legal report so much hue and cry and things are not there. But as the things are increasing, awareness are increasing, especially YouTube media, social media, the forensic medicine expert also started defensive practice. So yes, definitely, but by the expert analysis. Actually, I have published a paper on the medical legal interview, which encompasses many things. It is said that during my UG period, our teacher used to say that what are the steps of the diagnosis of disease. The one is history taking, second is the physical examination, and third is the investigation. If a doctor is expert in taking history and patient is also good in giving history, then more than 50% data can only be collected by the history. And physical examination 30%, and only 20% data can be collected by the investigation, different test. But, and same thing is in the crime investigation also. Same step, history, physical examination and investigation. Here 3 different agencies are working. For the history, police is working. For the physical examination, medical officers are working. And for the investigation test, here investigation means lab test, the forensic science laboratory people working. So history part is 50%, but definitely it should be taken by the expert person. So non-involvement of the expert at initial stage, like in medical examiner system of USA, their scientific person is more and more involved in the initial stage, so that at very initial stage case is concluded or case is narrowed and less investigation is required. So this model, nowadays the government of Bihar has started sending forensic science expert to the crime scene everywhere, thing. But still there is a less involvement. Crime scene investigation means the crime scene pe gaye forensic science experts they have some investigation collected and sample and entire thing. Later on, police officers are not in constant connection with the medical officer and scientific expert. So even in disease diagnosis, 50% data can be collected only when doctor is good. The person who is taking history is good. Similar-wise, here the investigation has to be done by the police personnel who is neither trained in the science nor trained in the law. So due to that, but definitely, and some company has created a safe kit and that safe kit all envelopes,
everything is
number of envelopes has to be inserted, fixed protocol. My opinion is always, protocol is made for the ideal situation and considering all the situation. This particular situation is, all situation cannot come in particular one situation. So your question is very right. That if from the history, very good information is available with the doctor, reliable information is available with the doctor. Doctor is also expert so that he can minimize the collection and then also less number of sample may come. But this is very ideal situation. Ideal situation, training में ये बता दे कि again we are doing the defensive practice. If that kit shall not be utilized completely, then again in court of law, some lawyers shall stand that, okay sir, your honor, this is the safe kit, this is the protocol, and only part of this protocol has been followed. So before making the protocol, at least some adjectives should be inserted in the protocol. These things shall be done in this situation when required, not in all as a blanket situation. So definitely but expert is required, and definitely by that convention, I think sample load can be reduced from our side. But we are also practicing the defensive medicine like doctors practicing the defensive medicine in that field. I think I have completed my answer.
Thank you.
To be very specific, as he said, in every case you got 20 samples, 20 into you examine 3 accused and you got 60. Now, I'll take a basic thing, we will do that. So, sample preservation, whose call is that? A doctor's call, who examines? And how does a doctor take that call? So, he examines the victim or survivor based upon that what evidential material need to be preserved. He takes a decision on that basis. Then there are certain recommended guidelines. Like if we go to Sehat guidelines and through which safe kit and everything came, and we also have ministry guidelines where they have a 17 long page documentation form for examination of survivor, and where they have listed so many 20-25 samples for that. Now, depending upon the case, this varies. If you get a case, based upon that, you decide what samples need to be preserved. For example, I'll just say, if there is history of oral sex, in that case, it is better you preserve oral samples. If it is not required, you don't have to preserve. But one of the aspect is that, like for example, what we practice for DNA sampling, suppose we'll take an example, what are the ideal samples, we know that. But it is also possible that if we preserve it, it may go for analysis, and it is possible also that we may not get result. So that's why we preserve at least 2 or 3 samples for DNA analysis. Example, I'm just taking it. I would very well correlate this with the samples which you preserve for toxicological analysis. एजेस से हम लोग 500 ग्राम का लिवर विद गॉल ब्लैडर, 10 ml ब्लड आचार जैसे हम लोग प्रिजर्व कर देते. देयर इज नो नीड. फोरेंसिक साइंस हैज इवॉल्व्ड सच दैट इफ यू गो अब्रॉड प्रैक्टिस करेंगे तो एक लिवर का टिश्यू रखते हैं, ब्लड रखते हैं, यूरिन रखते हैं, बस दैट्स इट. बट द गाइडलाइंस हैज टू बी कम फ्रॉम योर एंड, like on what basis? Because if you recommend these samples need to be preserved and it is enough for us. And practice of forensic varies at different aspect. If you take a person who is working in primary health center, who is a medical officer dealing with sexual assault cases or homicide cases, if you go to a medical officer who is working at a Sardar अस्पताल, आप higher tertiary case and medical college में भी practice करते, तो हर जगह पे practice थोड़ी सी अलग-अलग हो जाती है. अच्छा, मैं डॉक्टर हूँ, मैं डिसाइड कर लूँगा कि मैं सैंपल प्रिजर्व करूँगा. सपोज इफ इट कम्स, आई डोंट नो विद इन हाउ मच टाइम यू एनालाइज द सैंपल एंड व्हाट विल हैपन. इफ इट एनालाइज इमीडिएटली, आई थिंक यू विल गेट द रिजल्ट. आई एम टॉकिंग अ वेरी बेसिक प्रॉब्लम्स व्हिच वी फेस. Suppose if it comes negative, then ultimately I am the one who is going to get your report. I am the one who is going to interpret your report. And I am the one who is going to correlate with my medical examination findings. And I am going to give final opinion. And I am also going in the court of law to justify my opinion. So in this situation, the maximum sample, I'm just talking in general parlance. The practice again may, and that's why you are facing oversampling in that case. That is what I will take in that case. But I think you may have an answer on the situation कि what samples, if you get one simple, if आपको identify कर चुके हो कि DNA इसमें से, if you recover DNA exactly from teeth, I'm not sure what practices, and I think SOPs are there from DFSs as much as the protocol is concerned, including the format and that, because you practice with your all FSL practice with the base document of DFSs, if I'm not mistaken. Yeah, so I think that is a real situation where it lies. I think I am able to answer your question.
एक्यूज्ड अगर मान लीजिए किसी केस में सर 3 दिन बाद 4 दिन बाद पकड़ा जाता है, 1 या 2, ऐसे स्थिति में उसके सारे इंटरनल पार्ट्स के स्वैब लेने की क्या आवश्यकता है? हम ये कहते हैं कि आप एट अंडरवियर या जो भी इनर गारमेंट एट दैट टाइम एट द टाइम ऑफ क्राइम उसने पहना था वो आप दे दीजिए, और 4 दिन बाद 5 दिन बाद, वेयर इज आई थिंक इट इज नॉट
I
रिक्वायर्ड.
got your question, madam.
This
Only thing, the practice, वही तो बोल रहा हूँ कि practice हर जगह पे अलग-अलग रहती है. A person who has a person who is an expert in forensic medicine examining the accused, and a person who is a medical person may not have that much of knowledge about forensic medicine. उन दोनों में practice करने की different अलग-अलग हो जाती है. तो जो आने के बाद 1-2 दिन के बाद उसका क्या रिलेवेंस होगा और उसको जस्टिफाई भी करना है.
Sir.
बहुत बार अभी ना 375 IPC has been drastically changed. Earlier वो आते थे accused examination for 1 or 2 purpose objective. Now that has changed completely. और वो प्रैक्टिस की डिफर हो जाती है हर चीज के ऊपर, वेदर ही इज इन एज अ PHC वर्कर है, एज अ मेडिकल कॉलेज में कहाँ पे काम करे, उसके ऊपर डिपेंड रहता है कितना सैंपल प्रिजर्व करते हैं उसमें.
थैंक यू सर. सर ये कहेंगे हम अपने FSL सर से DFS सर की क्या ये हमें छूट होगी अगर हमारे पास 10-20 सैंपल अनावश्यक आ गए और हमें एक्यूज्ड के जैसे अंडरवियर मिल गया एट दैट टाइम क्राइम के समय पहना हुआ और उसके बाद अनावश्यक है तो हम उसको न करें, क्या ऐसा कुछ हो सकता है सर? नहीं मैम, कोर्ट में फिर प्रॉब्लम हो.
डॉक्टर शर्मा विल गिव द रिप्लाई हियर.
मे बी. हाँ सर.
क्या क्वेश्चन है?
सर मेरा ये क्वेश्चन है.
यस प्लीज.
कि जैसा मैंने पहले बोला कि बहुत सारे केसेस में एक्यूज्ड क्राइम में टाइम बहुत ज्यादा गैप होता है, द टाइम ऑफ क्राइम एंड जो एक्यूज से सैंपल मेडिकल में, तो उसमें अगर एक्यूज्ड के बहुत सारे सैंपल आ जाते हैं तो क्या ऐसा है कि हमें कोई ऐसा सैंपल जैसे उसके अंडरगारमेंट मिल गए जिसमें हमें रिजल्ट आ गया है जो भी विक्टिम और पूरी केस हिस्ट्री के अकॉर्डिंग, तो फर्दर उसमें जो अनावश्यक हमारे हिसाब से अनावश्यक है वो क्या हम 8-10 ऐसे सैंपल जो अलग अलग जैसे सर ने बताया वो विक्टिम के एस्पेक्ट में बताया, ये एक्यूज्ड के एस्पेक्ट में भी होता है कि अपर पार्ट से स्वैब, लोअर पार्ट फला सब कुछ होता है सर, तो क्या हम उसको ना करें? ऐसा कर सकते हैं क्या? और उसको नहीं करने की स्थिति में रिपोर्ट में हम क्या लिखेंगे सर?
मैडम सी
सर,
first the doctor should not have collected those samples.
Sir,
right? In case when the sexual assault has happened before 48 hours, there is no need to collect the accused penile swab and other
Yes, sir.
things.
Sir.
Garments are the separate thing.
Okay, sir.
Right? I'm thinking of the body sample. Right?
Right, sir.
And I'm sorry, I'm blunt actually. About 70 to 80% exhibits in biology and DNA are collected by the doctors. Right?
Right, sir.
I had a case which I did not accept, right, where the lady eloped with someone 6 months ago. After 6 months, she filed an FIR and her vaginal swab was collected and sent for forensic examination. It should not have been collected.
सर हमारे ऐसे
And
बहुत
then,
केस आते
see,
हैं.
I have received cases where identity of deceased is there, dead body is there. These 10 digits of the finger, these digits were cut and sent to me for DNA profiling.
Sir.
Right? And I think many books are written by the foreigners, medical jurisprudence books are there, right? And same are being followed by our authors, our Indian authors. But the practical situation which we discussed in the morning also with Dr. Devinder Kumar, the quality of alcohol in hospital is very, very poor, right? It has lot of inhibitors. You have a salt solution that is perhaps the best solution for the safe, right? So there are many things, and again we are receiving the exhibits preserved in formalin. Fetus is preserved in formalin, right? And when we reject the case, they simply strike it out, the formalin, and overwrite it with the salt solution.
Yes, sir.
Right, we have to accept it. So these are some of the situations, right? Perhaps there should be a more interaction with the forensic biologist and the medico-legal personals, right?
Right, sir.
And CFCL Calcutta started the DNA profiling in 97. So we received cases from the whole country and Nepal and Bhutan. So and another thing, I don't know which medical book or the jurisprudence book has written that in sexual assault case, accused semen has to be collected. I don't know. Yeah,
So
right.
many steps.
I don't know.
Steps are taken.
See, his semen has to be collected. I don't know which book has written it. I have written to the health ministry also, but I did not get any reply regarding that. And since the CDTS is below our ground floor in the CFCL Calcutta, right? This fact was told to me one IO from the Haryana. He said that you will say that the semen on the petticoat of victim is of the accused. Do you know how it is planted? Whether you can say it's planted or not, no, I cannot say. No. I can say only the individualization, he is from this person. He said the doctor collects the sample of the accused and police or any other agency simply plant on the relevant exhibits, vaginal swab and other thing. Right. So then it comes to you. You have to identify it. There are many gaps. Many gaps. Yeah, please.
No.
Okay. So actually, my point of view in this case is that being a forensic expert, when we examine a case, what are the parameters that we should taken care of? Many times, many samples they land in a particular case. Yes, I understand. But being a scientist in the lab, our focus should be the questions which are asked by the investigating agency, which is written in the draft, that should be answered. So we are having a very limited role when we are working in the lab. That question should be reframed, because you see, what should be the objective of an examination? That we need to understand. What is the objective? When the investigating agency is investigating a case, what is its objective? Objective is, maybe in a case of sexual assault, to identify the culprit. Culprit identification can be done in both the ways, identifying the biological sample of the victim on the suspect's body or vice versa. It can be done in either of the ways. So we need to train the investigating agency also to frame the questions. If the questions can be framed properly, then we will be able to work accordingly in the lab to get the answer. Because our role is very difficult, because if they have put 5 questions and we are bound to answer all the answer for these 5 questions, we cannot escape any one of them. Otherwise, the defense lawyer can ask us why you have not given answer on so point. So our role, being in the laboratory workers, bench workers I am talking about, particularly those who are reporting officers, our work is much limited. So training should be done extensively. Yes, those who are working in the forefront when I am talking about the investigating agency, definitely after the implementation of new criminal law, I hope when the forensic experts, they will land on the in the crime scene during the collection of evidences, it will change the scenario. It will change the scenario. But as I was saying, forensic expert, the role is very much limited. It is 20%. Majority of the samples are coming from the hospitals during either medical legal examination or post-mortem examinations. So there should be a cohesive interaction, fruitful interaction among all the fraternities, so that a conclusion can be done over here. Thank you.
Sir, from my side, there is my humble request to Chief Forensic Scientist, sir.
Just I want to add in whatever Hirak Ranjan Das was saying. इस चीज को देखते हुए ना, इसीलिए फोरेंसिक एक्सपर्ट का क्राइम सीन पे जाना शुरू किया गया है. अभी फोरेंसिक एक्सपर्ट के पास ये डिस्क्रिशन है एट क्राइम सीन, because he is doing tests also कि सीमन कहाँ है कहाँ नहीं है, तो क्राइम सीन के एविडेंसेस तो वो लिमिट कर सकता है कि जो कलेक्शन है वो सही से हो, मतलब मल्टीपल सैंपल्स नहीं हो. पुलिस पहले क्या करती थी कि जो भी एविडेंसेस मिलेंगे सारे कलेक्ट कर लेंगे वो, है ना. अभी फोरेंसिक एक्सपर्ट क्राइम सीन पे जाता है तो वो रेलेवेंसी, जो भी रेलेवेंट एग्जिबिट्स हैं वो कलेक्ट कर सकते हैं वो, और प्रिजर्वेशन भी उसका सही से होगा. ये अभी लॉ में भी है तो इसलिए 176 में कवर्ड है, तो ये चीज का ध्यान दिया जा सकता है. दूसरा, जो एक्सपर्ट है उसको भी ये डिस्क्रिशन है कि रेलेवेंट एग्जिबिट्स कर सके वो. अगर कोई स्केलेटल रिमेन में 50 एग्जिबिट्स आए हुए हैं तो आइडेंटिफिकेशन में रैंडमली 4 अगर वो करते हैं और वो मैच करता है पेरेंट्स के साथ, क्लेमेंट्स के साथ, तो वो 45 को छोड़ सकते हैं. ऐसे ही मेरा एक्चुअली क्वेश्चन आपसे है कि एक कुछ टाइमलाइन फिक्स करनी चाहिए हमें कि 6 महीने पहले जैसे रेप हुआ है कोई, तो 6 महीने के बाद रिपोर्ट करते हैं वो, उनके सारे एग्जिबिट्स क्यों कलेक्ट किए जाते हैं? जैसे सैक किट है, ठीक है, तो कुछ ऐसा मतलब प्रोविजन होना चाहिए कि वो अवॉइड किया जा सके.
Sir, my request is for you, sir. Actually, I am assistant director in RFSL Bhagalpur, and we are having infrastructure for DNA setup. 2,400 square feet laboratory is built and infrastructure is there. Sanctioned posts are there. Manpower is there. 2 assistant director who did their doctorate from RMRI Patna, and they have 4 or 5 year knowledge of DNA analysis are there. Scientific assistants are there, but we are not having machine. Sir, it is the request.
You will get very soon.
Sir, it will be huge grace from your side, sir.
In advance, you will get very soon. Okay.
Thank you, sir.
So, what's the question?
Actually, sir, no doubt what you have said that it is not useful, but this is the formality. Many things in the crime investigation, people do it for formality. And everybody knows, even doctors knows, this is useless sample. But definitely it should be reduced. And one suggestion which came from, you are from the UP, I think I had met you in the director's chamber of the FSL, UP, Dr. Rajas, okay, thank you. So what we practice in medical science, when we send samples for the histopathology. So when we in our histopathology department, so clinician collect the whole heart, whole lung, or whatever the organ they have to send. It is in the laboratory. There are various screening processes first. In the histopathology lab, various steps are there. They do grossing. And by that screening process, by that grossing process, our pathologists decide that from which portion sample has to be taken and it has to be processed. They are the person who can decide. And accordingly, because the clinician, whether in surgery, medicine, gynecology, whoever there, or we are in forensic medicine clinician of dead bodies, we shall collect the whole heart, whole lung, etc. and we send for the histopathology lab, and they screen it. They identified that which portion is important and which portion is not important, and accordingly do histopathology. So this power already sir has also said, and this discretion is also already vested in you. You are the expert, and you can decide that which is relevant sample and which is not relevant sample. So I think sir has also stated the same thing. So it is already, I think, in the crime investigation also, investigating officer collects all the cloths. Suppose for semen analysis, there by the UV ray they screen, and inside the laboratory ultraviolet they screen the site and identify the site. And where the site is not identified, it is left. It is not necessary to examine the entire portion of the cloth. I think same thing is applicable in these samples also.
Now, because time is very short and we will move very fast to the next session of valediction, but Still, I have some suggestion or some queries in my mind because a lot of experts from the country are here. Regarding the equipment for your lab in Bihar, we will give very soon, and your proposal is under consideration. And Dr. Binay Kumar told regarding the PPP model. I want to give one clarification here. Regarding PPP model, we will only taking this PPP model, they will put the equipments, they will put the chemicals, and they will give only the experts to run the equipment. Other things will be in the purely control of the experts. They have no control over it. Reason is, the 50% of the time of our experts is going on arranging all these things. So they will save some time, and these things are immediately available to them, and there will not be a problem that these are the financial procedure and all. Those things can be minimized and it can be worked. This is a step-up arrangement only. After full-fledged development of the laboratories, full-fledged manpower, these things can be separated from the lab. Regarding the procedure adopted in the labs, procedure adopted in the medical examination, there is an urgent need, as Dr. Already told, there is a need of training. Training and retraining is very much important, and because this aspect is really handled very casual manner in our labs, also in the hospital also. But this is the urgent need. This training and retraining should be there. And as a DFSS, we have prepared the 7 clusters of our CFSLs, and they will provide the training of their state FSL scientists, and simultaneously they will also give the training to the medical doctors, awareness programs. Similarly, the state FSLs should also give the training to the regional FSL scientists as well as the doctors in their areas, so that it can be uniform, it can be work in totality, and there will be minimum chances of mistake. This was the idea, and we have also issued the order. Similarly for Guwahati, they will take care of northeastern labs. For Pune, Bhopal and Guwahati and Delhi, we have already given, and those directors are taking care. This is regarding training is very important. Now the SEC kit, why we have prepared the SEC kit? Reason is, sometimes the police officers is not able to collect the samples properly, and it's a mixed sample. So it has no use. When it is mixed, properly contaminated, and laboratory is not able to give the report. On the direction of the WCD ministry, we prepared that kit and it distributed to all police officers, this kit. And our forensic experts will go. As Dr. Sharma told, in cases where punishment is 7 years or more, under 176(3), the forensic scientist visit is compulsory. It is mandatory. No doubt we have the limited number of the scientists, but we have to go positively. Though the IO is the custodian of the crime scene, he is the custodian, but we have to go, we have to identify the evidence, we have to collect it, properly pack it, hand over it, and seal it. Because longitude and latitude is also important. Sometimes the exhibit is collected, say in UP, in say Lucknow, and it is shown as Kanpur, like this. This should not happen. Similarly in ganja cases or narcotics cases, and we have so we have under e-Saksh, there is a need of putting all these information, date and time, longitude, latitude, and name of the police officers, name of the thana, and it will be printed on the exhibits, just like a path lab. So that when this sample go to the lab, they will scan it, everything will be pop up. So ICJS, interoperable criminal justice, is also in place, and I think more than 50% lab are using ICJS 2, and around 31 labs are using ICJS 1. So very soon all the labs should come on ICJS 2 also, and report will go online. These are some initiatives taken by the Ministry of Home Affairs, And regarding the preservation of evidence and all this, these are the part of training part, I think. And when you will interact with the scientists or doctors, then it will automatically close the chapter. So because time is very short, I am not taking much time. If you have any questions, the panelists are here. Otherwise, we will close it. Thank you. If you have any questions, please. Any question from your side? Or suggestion? The forensic science? Any suggestion or question from your side? If not, then we can close it. Okay, thank you very much. Yeah, you have. Yeah, please.
Sir, in Jharkhand, there is a bovine act. वहाँ पे काऊ का स्लॉटर करना प्रोहिबिटेड है, तो वहाँ पे कैटल मैमिंग केसेस से काऊ का प्लेस आता है हम लोग का लैब में, तो उसका सीरोलॉजिकल टेस्ट करके हम लोग देते हैं। तो सर उसमें हम लोग क्या वैलिडेटेड कोई किट है ऐसा जो इंडिया में यूज होता है जो अप्रूव्ड हो कि, तो सर हम लोग अपना लैब में उसको इंट्रोड्यूस करना चाहेंगे सर, उससे हमारा टाइम भी बचेगा और एक्यूरेट रिजल्ट भी जाएगा। हाँ, तो वो हम लोग अपना लैब में इंट्रोड्यूस करना चाहते हैं, तो वो कैसे हम लोग करेंगे?
RT-PCR किट है,
जी जी,
हाई मीडिया का भी है आप?
हाँ सर, वो हम लोग के यहाँ डेमो भी हुआ है, बट हम लोग उसको इंट्रोड्यूस कैसे करें वो हम लोग नहीं कर पा रहे अभी। हम लोग का हाँ नहीं, उसको अपने लैब में इंट्रोड्यूस करना है सर, तो उसका गाइडलाइंस अगर अप्रूवल है उसका DFSL से, हम लोग क्यों नहीं कर पा रहे? हम
ऐसा कुछ नहीं है। जो RT-PCR हम HID सॉफ्टवेयर यूज कर रहे हैं ह्यूमन आइडेंटिफिकेशन के लिए, उस ढंग से हम एनिमल्स का भी आइडेंटिफिकेशन कर सकते हैं। उसका अपना केमिस्ट्री है, प्लस हाई मीडिया के भी कई एंटीसीराज अवेलेबल हैं मार्केट में। अब कमर्शियल एंटीसीराज भी अवेलेबल हो रही हैं।
सर अगर DNA बेस्ड है, अगर वो डेमो भी हुआ है बट वो इंट्रोड्यूस नहीं हो पा रहा है हम लोग के यहाँ।
आप ना उसका इंटरनल वैलिडेशन कर लो, कुछ पहले कंट्रोल सैंपल्स लेके ना, जब वो आपको रिप्रोड्यूसिबल रिजल्ट देना शुरू कर दे तो उसको इम्प्लीमेंट कर दो।
अगर DFSL से अप्रूव्ड है तो सर हम लोग इंट्रोड्यूस कर लेंगे उसको। RT-PCR तो है मैडम, ठीक है, तो ठीक है मैडम, ठीक सर, ओके। थैंक यू सर।
By this, we end today's fifth session and move toward the valedictory of our two-day satellite conference, Setcon 2026. Thank you.
आधा टेंशन नहीं चलता।
We will begin with the valedictory session for this conference very shortly. All are kindly requested to please remain seated.
प्रोग्राम का हो जाएगा प्रोग्राम का प्रोग्राम का होगा सीक्वेंस सब जानकारी कंफर्म कर लीजिए कि क्लास कुछ नहीं है मीडियम हम दिखा दिए ठीक है बैठिए क्लास करते हैं
ये डिस्प्ले चाहिए इसको आप पे आप नहीं है ये वाला डिस्प्ले उसमें नहीं लग गया डिस्प्ले होते रहेगा ये सिस्टम यहाँ नीचे पानी का बॉटल यहाँ पे ऐसे रहे यहाँ से
मेरी सभी से रिक्वेस्ट है कि वो फीडबैक फॉर्म सेकेंड डे का भर के हमारे वॉलंटियर्स को हैंड ओवर कर दें प्लीज।
कुछ ही क्षणों में हमारे बीच आज के मुख्य अतिथि का आगमन होने जा रहा है। अब हमारे बीच उपस्थित हैं आज के मुख्य अतिथि श्री अरविंद कुमार चौधरी सर, अपर मुख्य सचिव गृह विभाग बिहार सरकार। आदरणीय मुख्य अतिथि श्री अरविंद कुमार चौधरी, अपर मुख्य सचिव गृह विभाग बिहार सरकार, मंचासीन विशिष्ट अतिथिगण तथा यहाँ उपस्थित सभी सम्मानित प्रतिनिधि एवं प्रतिभागी, इस 2 दिवसीय सैटेलाइट सम्मेलन के समापन सत्र में मैं आप सभी का स्वागत करती हूँ। आज के इस समापन सत्र के मुख्य अतिथि श्री अरविंद कुमार चौधरी सर का एक संक्षिप्त परिचय। सर 1995 बैच बिहार काडर के भारतीय प्रशासनिक सेवा के एक वरिष्ठ अधिकारी हैं तथा वर्तमान में बिहार सरकार के गृह विभाग में अपर मुख्य सचिव के पद पर पदस्थापित हैं। इस महत्वपूर्ण दायित्व का निर्वहन करते हुए सर राज्य की सुरक्षा एवं विधि व्यवस्था से जुड़े प्रमुख विषयों का कुशलतापूर्वक संचालन कर रहे हैं। इसके अतिरिक्त 31 अगस्त 2025 से सर मंत्रिमंडल सचिवालय विभाग का अतिरिक्त प्रभार भी संभाल रहे हैं, जहां ये उच्च स्तरीय प्रशासनिक समन्वय में महत्वपूर्ण भूमिका निभा रहे हैं। अपने समृद्ध प्रशासनिक अनुभव के दौरान सर ने वित्त विभाग में प्रधान सचिव के रूप में राज्य की वित्तीय नीतियों एवं बजट प्रबंधन को दिशा दी तथा ग्रामीण विकास विभाग में प्रधान सचिव के रूप में आधारभूत संरचना एवं गरीबी उन्मूलन से जुड़े महत्वपूर्ण कार्यक्रमों का सफल नेतृत्व किया। साथ ही बिहार ग्रामीण आजीविका संवर्धन सोसाइटी जीविका के मुख्य कार्यपालक पदाधिकारी के रूप में सर ने 7 वर्षों से अधिक समय तक कार्य करते हुए इसे 80 लाख से अधिक ग्रामीण परिवारों तक पहुंचाने वाले एक सशक्त जन आंदोलन का रूप दिया। ग्रामीण आजीविका के क्षेत्र में इनके उत्कृष्ट योगदान के लिए वर्ष 2011 में सर को प्रतिष्ठित स्कॉच चैलेंजर्स अवार्ड से सम्मानित किया गया। महिला सशक्तिकरण एवं वित्तीय समावेशन के क्षेत्र में सर के नेतृत्व में बिहार देश का पहला राज्य बना जहां 10 लाख से अधिक महिला सेल्फ हेल्प ग्रुप्स स्थापित किए गए। सूचना प्रौद्योगिकी के क्षेत्र में भी सर ने नवाचार को बढ़ावा देते हुए ड्रोन जैसी उभरती तकनीकों के उपयोग को प्रोत्साहित किया, जिससे पारदर्शिता एवं निगरानी में उल्लेखनीय सुधार हुआ। सर का प्रशासनिक नेतृत्व उत्कृष्टता, नवाचार एवं जन कल्याण के प्रति इनकी गहन प्रतिबद्धता को प्रतिबिंबित करता है। सर, हम आपकी गरिमामयी उपस्थिति के लिए हृदय से आभार व्यक्त करते हैं। मैं अनुरोध करती हूँ श्री हिमजय कुमार, निदेशक विधि विज्ञान प्रयोगशाला पटना से, कि वे हमारे मुख्य अतिथि श्री अरविंद चौधरी सर को पौधा भेंट कर उनका स्वागत करें। सर, बिहार में पहली बार यह ऐतिहासिक 2 दिवसीय सैटेलाइट सम्मेलन सेटकॉन 2026 का आयोजन किया गया। पिछले 2 दिनों में देश विदेश से आए प्रतिष्ठित वैज्ञानिकों एवं प्रतिनिधियों की सक्रिय एवं उत्साहपूर्ण सहभागिता देखने को मिली। इस सैटेलाइट सम्मेलन के दौरान डीएनए विश्लेषण, फॉरेंसिक विज्ञान के विभिन्न आयाम, फ्यूचरिस्टिक बायोटेक्नोलॉजी, एआई के युग में फॉरेंसिक की भूमिका तथा डेटा संरक्षण जैसे अत्यंत समकालीन एवं महत्वपूर्ण विषयों पर गहन विचार विमर्श, ज्ञान साझाकरण एवं सार्थक संवाद किया गया। पिछले 2 दिनों में विभिन्न टेक्निकल सेशंस में ओरल पेपर प्रेजेंटेशंस और पोस्टर प्रेजेंटेशंस प्रतियोगिता भी आयोजित की गई। मैं अब हमारे माननीय मुख्य अतिथि महोदय से विनम्र अनुरोध करती हूँ कि वे हमारे प्रतियोगिताओं के विजेताओं को पुरस्कृत करने की कृपा करें। सबसे पहले ओरल पेपर प्रेजेंटेशन इवेंट में प्रोफेशनल्स कैटेगरी में 3rd पोजीशन जाता है अजय कुमार राणा, साइंटिस्ट बी, CFSL हैदराबाद को। उनका टॉपिक था सिबलिंग आइडेंटिफिकेशन from 2 decades old skeletal remains using combined autosomal SIBSHIP analysis and Y chromosomal STR profiling, a case study. सेकेंड पोजीशन जाता है श्रीमती एमी, असिस्टेंट डायरेक्टर FSL मिजोरम। इनका टॉपिक था हिडन एविडेंस इन लॉन्ड्री, STR DNA profiling of washing machine lint in a homicide investigation. फर्स्ट पोजीशन इन ओरल पेपर प्रेजेंटेशन जाता है डॉक्टर स्वीटी शर्मा, प्रिंसिपल गीता यूनिवर्सिटी पानीपत हरियाणा को। टॉपिक अ स्पेक्ट्रोस्कोपिक एंड कीमोमेट्रिक अप्रोच इन फोरेंसिक साइंस फॉर द डिस्क्रिमिनेशन ऑफ एंटीमॉर्टम एंड पोस्टमॉर्टम ब्लड। अगला इवेंट पोस्टर प्रेजेंटेशन, इसमें प्रोफेशनल्स कैटेगरी में 3rd पोजीशन जाता है मिस सुनीता वर्मा, डेप्यूटी डायरेक्टर CFSL पुणे को। टॉपिक फर्स्ट क्रॉस बॉर्डर ह्यूमन आइडेंटिफिकेशन फ्रॉम इंडिया यूजिंग इंटरपोल्स डिजिटल DNA डेटा एक्सचेंज, अ लैंडमार्क फोरेंसिक केस स्टडी। सेकेंड पोजीशन पोस्टर प्रेजेंटेशन में जाता है मिस सीमा डागर, CFSL न्यू दिल्ली को। टॉपिक 29 years mystery of green boots resolved via DNA test। फर्स्ट पोजीशन जाता है श्री नीरज कुमार वार्ष्णे, असिस्टेंट डायरेक्टर FSL पटना को। टॉपिक डबल मर्डर केस मिस्ट्री सॉल्व्ड बाय बायोलॉजिकल क्लू मटेरियल, अ केस स्टडी। हमारे स्टूडेंट कैटेगरी में पोस्टर प्रेजेंटेशन में विनर हैं निलंजन साहा, टॉपिक persistence and recovery of human environmental DNA (eDNA) from aquatic environment after submersion of saliva stained fabrics. इसी के साथ हमारा पुरस्कार वितरण सत्र समाप्त होता है। सभी विजेताओं को हार्दिक बधाई। अब मैं निवेदन पूर्वक आमंत्रित करती हूं हमारे मुख्य अतिथि श्री अरविंद कुमार चौधरी सर को कि वे इस सैटेलाइट सम्मेलन के समापन सत्र को संबोधित करें।
एडीजी सीआईडी बिहार श्री पारसनाथ जी, एसके जैन सीएफएस डीएफएसएस न्यू दिल्ली, बीके महापात्रा जी जो सीएफएसएल दिल्ली से आए हैं, उसके निदेशक हैं, जयंत कांत जी, हिमजय जी, इस 2 दिवसीय कॉन्फ्रेंस में भाग लेने वाले विभिन्न राज्यों से आए प्रतिभागीगण और फॉरेंसिक साइंसेज से जुड़े विभिन्न इंडस्ट्री से आए प्रतिनिधिगण, मैं सबसे पहले पारसनाथ जी को और एसके जैन साहब को बहुत बहुत धन्यवाद देता हूं कि आपने संयुक्त रूप से यह पहल की और मुझे बताया गया कि बिहार में कई दशकों के बाद इस तरीके का कॉन्फ्रेंस हुआ है। मैं सीआईडी की और फॉरेंसिक साइंस की इनकी पूरी टीम को भी इसके लिए बधाई देता हूं कि आपने ये दोनों दिनों का कार्यशाला बहुत ही अच्छे तरीके से आयोजित की, इस कॉन्फ्रेंस को बहुत अच्छे तरीके से आयोजित किया। नए आपराधिक कानूनों के आने के बाद से फॉरेंसिक साइंस का महत्व और भी ज़्यादा बढ़ गया है। यद्यपि पहले भी इसका महत्व था, लेकिन अब तो यह अपेक्षा है कि ज़्यादातर जो इन्वेस्टिगेशन हो वो साइंटिफिक तरीके से हो। उसके लिए बहुत ही आवश्यक है कि हमारे पास अच्छे लैब्स हों, एक अच्छा और वेल क्वालिफाइड मैन पावर हो और हम समय पर इन्वेस्टिगेशन में या न्यायालय में जहां पर कि उस मामले की सुनवाई चल रही है उसमें मदद कर सकें। यह एक बहुत बड़ा गैप है। मुझे याद पड़ता है कि 3 कानूनों के संबंध में एक बहुत बड़ा कॉन्फ्रेंस हुआ था जिसमें पटना उच्च न्यायालय के माननीय मुख्य न्यायाधीश भी आए थे। उस दिन एक न्यूज़पेपर में फुल पेज का एक रिपोर्ट छपा था कि अलग अलग राज्यों में क्या कैपेसिटी है फॉरेंसिक के मामले में, और ख़ासकर नए आपराधिक कानूनों के आलोक में इसके बारे में चर्चा की गई थी। और बिहार उसमें काफ़ी पीछे था, और कुछ राज्य जिसको आगे दिखाए जा रहे थे, कर्नाटका हो गया, इवन केरल है, तमिलनाडु है, इसको उन्होंने दिखाया। संयोग की बात है कि उस दिन मैंने भी सुबह में वो देखा था, नोटिस किया था, और उसकी फ़ोटो मेरे मोबाइल में भी मौजूद थी। तो मैंने उनको कहा था कि सर हम लोग लगातार प्रयास कर रहे हैं, और आपको अभी यहाँ हमारी टीम ने बताया भी होगा कि हमारे FSLs अब कितने फंक्शनल हैं, कितने और FSL पाइपलाइन में हैं जो कि नए सिरे से डेवलप होने जा रहे हैं। बिल्डिंग्स लगभग तैयार है, संभवतः 1 साल के अंदर अंदर हम उनको फुल्ली फंक्शनल कर लेंगे। इनकी नियमावली में संशोधन अंतिम चरण पर है। नियमित नियुक्ति होने तक के गैप को फुलफिल करने के लिए बिहार ने कॉन्ट्रैक्ट पर ऑलरेडी काफ़ी लोगों को नियुक्त कर लिया है। 103 लोगों को नियुक्त किया है, और इन सब से काफ़ी ज़्यादा मदद मिलेगी। मोबाइल फॉरेंसिक वैन भी हमारे पास काफ़ी आ गए हैं, हर ज़िले में कार्यरत हैं। ये निश्चित रूप से हमें मदद करेगा आगे जाने के लिए। लेकिन ये भी बात बहुत महत्वपूर्ण है कि जिस तरीके से तकनीक बदल रही है, हम नई तकनीक की भी जानकारी लगातार प्राप्त करते रहें। मुझे अच्छा लगा कि आपने जो प्रतियोगिता आयोजित की थी वो पूरे देश के स्तर पर आयोजित की थी, और इसमें विभिन्न स्तर के लोगों ने भाग लिया है, बच्चों ने भी भाग लिया है, युवाओं ने। तो ये उनके बीच भी एक साइंटिफिक टेम्पर डेवलप करने का, फॉरेंसिक से रिलेटेड चीज़ों के बारे में एक समझ डेवलप करने का बहुत अच्छा प्रयास है। मैं आप सब लोगों को इसके लिए बहुत बहुत बधाई देता हूँ। जिन विषयों पर चर्चा हुई है, यहाँ पर फॉरेंसिक बायोलॉजिकल साइंसेस पर यह कॉन्फ्रेंस था। इससे रिलेटेड जिन विषयों पर चर्चा हुई है, मैं पा रहा हूँ कि वे बहुत ही सामयिक हैं, कंटेम्प्रेरी हैं, और बहुत ही उपयोगी हैं। ये भी मैंने देखा कि बहुत सारी एजेंसीज़ ने भी अपने जो नए नए प्रोडक्ट्स हैं उनको यहाँ पर डिस्प्ले किया है, और ये दिखाया है कि कैसे हम उनका उपयोग करके और बेहतर तरीके से जांच कर सकते हैं। मुझे पूरा यकीन है कि इन चीज़ों का हम फ़ायदा उठाएंगे। भारत सरकार का गृह मंत्रालय भी राज्यों की फॉरेंसिक कैपेसिटी को मजबूत करने में, उसको आगे बढ़ाने में लगातार प्रयास कर रहा है, और जैन साहब की यहाँ उपस्थिति से मुझे पूरा विश्वास है कि हमें उनका और गृह मंत्रालय का पूर्ण सहयोग मिलेगा। हम लोगों ने अपनी जितनी समझ थी उसके हिसाब से एक प्रस्ताव ऑलरेडी भेजा हुआ है, जो एक उनके स्तर पर, उच्च स्तर पर अभी विचार विमर्श में है। मैं उम्मीद करता हूं कि जल्दी उस पर हमें स्वीकृति मिलेगी, और आपके गाइडेंस में हम लोग भविष्य की हमारी क्या कार्य योजना होगी उसके आलोक में नए सिरे से और भी हमारी जो रिक्वायरमेंट हैं उसके हिसाब से अपना प्रस्ताव तैयार कर आपको भेजेंगे। मुझे ये बताते हुए भी खुशी है कि बिहार में नेशनल फॉरेंसिक साइंसेज यूनिवर्सिटी की भी एक शाखा खुल रही है, उसका बिहार में भी एक ब्रांच खुल रहा है, और उसके ही कैंपस में CFSL की भी एक यूनिट को लोकेट होगी। इसके लिए गृह मंत्रालय, भारत सरकार ने स्वीकृति दे दी है। राज्य सरकार ने भी इसके लिए ज़मीन इत्यादि चिन्हित कर लिया है। मंत्रिपरिषद की स्वीकृति के बाद भूमि फॉर्मली हस्तांतरण की कार्रवाई की जाएगी। इस बीच में एक टेम्पररी ट्रांजिट कैंपस आइडेंटिफाई कर लिया गया है, उसको शिफ्ट करके हैंडओवर करने की कार्रवाई की जा रही है। तो मैं उम्मीद करता हूं कि आगामी सत्र जो जुलाई से प्रारंभ होगा उसमें बिहार कैंपस में भी पढ़ाई या कुछ कोर्सेज शुरू हो सकेंगे, ये मैं उम्मीद करता हूँ। तो आप सबको जो भाग लेने आए हैं बिहार में आने के लिए, पटना में आने के लिए बहुत धन्यवाद और आपका स्वागत मैं करता हूँ। मैं ये भी उम्मीद करता हूँ कि अगर आप थोड़ा समय लेकर आए होंगे तो, और अगर कहीं आपकी ये पहली विजिट होगी तो जो अच्छी जगह हैं यहाँ की उनको देखकर भी आप जा सकते हैं। बिहार का जो बिहार म्यूज़ियम है ये वाकई वर्ल्ड क्लास म्यूज़ियम है और अभी विदेशों से भी जितने भी लोग आते हैं, कई कई ऐसे लोग हैं जो 6 महीने 8 महीने पहले से अपना प्रोग्राम बनाते हैं और उसमें काफ़ी ज़्यादा समय यहाँ म्यूज़ियम में बिताते हैं। ये म्यूज़ियम वाकई में इसने काफ़ी सारे अंतर्राष्ट्रीय पुरस्कार जीते हैं और जिस किसी को भी हमने भेजा है वहाँ पर, तो जाने के बाद उनका यही रिमार्क होता है कि हमने नहीं उम्मीद की थी कि इतना बढ़िया म्यूज़ियम बिहार में होगा। एंड इट इज़ डेफिनेटली नॉट ओनली द बेस्ट इन द कंट्री बट परहैप्स इवन इन द एंटायर साउथ ईस्ट एशिया, वर्ल्ड में भी चौथे 5वें नंबर पर यह म्यूज़ियम होगा। तो आप लोगों ने अगर उसको नहीं देखा है तो मैं ज़रूर आपको सुझाव दूंगा कि आप उसको एक बार देख लें। उसके अलावा भी बहुत सारी चीज़ें हैं, हमारी टीम है आपको गाइड कर सकेगी इसके लिए। तो एक बार आयोजकों को फिर से इस कार्यशाला के, इस कॉन्फ्रेंस के आयोजन के लिए बधाई देते हुए अपनी तरफ से बिहार में फॉरेंसिक की कैपेसिटी को और बढ़ाने में पूरा सहयोग देने के आश्वासन के साथ मैं अपनी बात समाप्त करता हूँ। धन्यवाद।
मैं अनुरोध करती हूँ श्री हिमजय कुमार, निदेशक विधि विज्ञान प्रयोगशाला पटना से, कि वे हमारे मुख्य अतिथि का धन्यवाद ज्ञापन करें।
आदरणीय अपर मुख्य सचिव श्री अरविंद कुमार चौधरी सर, एडीजी सीआईडी श्री पारसनाथ सर, डीआईजी सीआईडी श्री जयंतकांत सर, डॉक्टर एसके जैन सीएफएस डायरेक्टरेट फॉरेंसिक साइंस एमएचए सर, डॉक्टर बीके मोहपात्रा निदेशक सीएफएसएल नई दिल्ली सर, डॉक्टर एके शर्मा सर सीएफएसएल गुवाहाटी निदेशक, एवं सभी गणमान्य अतिथिगण जो विभिन्न विधि विज्ञान प्रयोगशालाओं, केंद्रीय विधि विज्ञान प्रयोगशालाओं एवं छात्रगण जो विभिन्न विश्वविद्यालय एवं उनके प्रोफेसरगण, मैं सबका धन्यवाद
असम श्री A.K. शर्मा सर को स्मृति चिन्ह भेंट करने हेतु मैं आमंत्रित करती हूँ। श्रीमती अजरा कमाल, असिस्टेंट डायरेक्टर FSL पटना, बिहार। SP CID श्री आदित्य कुमार को स्मृति चिन्ह भेंट करने के लिए मैं आमंत्रित करती हूँ। सीमा पटेल, असिस्टेंट डायरेक्टर FSL पटना, बिहार। श्री हिमजय कुमार, डायरेक्टर FSL पटना को सम्मानित करने के लिए मैं आमंत्रित करती हूँ श्री जितेंद्र कुमार, असिस्टेंट डायरेक्टर FSL पटना, बिहार को। हमें विश्वास है कि इस सम्मेलन में प्राप्त अनुभव, विचार एवं निष्कर्ष आने वाले समय में फॉरेंसिक विज्ञान और आपराधिक न्याय प्रणाली को नई दिशा प्रदान करेंगे तथा हमारे कार्य क्षेत्र में उत्कृष्टता को प्रोत्साहित करेंगे। इसी के साथ इस सैटेलाइट सम्मेलन का समाप्ति का ये उद्घोषणा मैं करती हूँ। आप सभी को बहुत धन्यवाद एवं शुभकामनाएं। अंत में हम जनगण मन और नेशनल एंथम के लिए आप सभी से अनुरोध करते हैं कि आप सभी अपने सीट्स पे खड़े हो जाएं। जनगण मन अधिनायक जय हे भारत भाग्य विधाता। पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंगा। विंध्य हिमाचल यमुना गंगा उच्छल जलधि तरंगा। तब शुभ नामे जागे, तब शुभ आशीष मांगे। गाहे तब जय गाथा, जनगण मंगलदायक जय हे भारत भाग्य विधाता। जय हे जय हे जय हे जय 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 हे। जय हिंद। आप सभी के पार्टिसिपेशन सर्टिफिकेट्स रजिस्ट्रेशन डेस्क से कलेक्ट कर सकते हैं और आप लोगों की रिफ्रेशमेंट्स के लिए भी व्यवस्थाएं। धन्यवाद।